Uh, I first have to apologize for I was so looking forward to being there in that room with you, delivering this talk, and things just did not work out that way. Uh, I apologize, but I will get there at some point. And I wish I was there as my three other founding members of HSI. It was 40 years ago that we launched this thing, and I'm so pleased how far it's gone. I didn't know what the really the big interest in heliconias and zingiberales were at that point, particularly commercially and particularly horticulturally. And HSI really demonstrated that they have a big following around the world. We started with Heliconia and then quickly expanded to the other seven families in the zingiberales. So I think we've shown that they are an important group of plants, tropical plants, but that are appreciated by many. I want to thank the organizers and particularly the speaker group for inviting me to give this keynote. It's interesting. Carla points out the first bulletin, which has abstracts of the first papers given in our conference in 1985. And I gave a paper there called, let's see, what was it? An Introduction to the Taxonomy and Classification of Heliconia. And here, 40 years later, I'm finally have some of the work done to be able to give you a definitive classification to the of Heliconia and also some important information on its conservation. So we've made uh, progress. Let me also mention that it's snowing here in Vermont. So I hope that makes you all feel better being there in Manoa. I wish I was there, as I said, with you. So let's begin. I'm going to share my screen, talk for maybe 40, 45 minutes about Heliconia and conservation, and then hopefully we'll have a discussion and some questions after that. I don't think I can see you too much after this, so let's just see how it goes. Okay. All right, I assume that people can see this on the screen there, Russ?
Yes, looks great.
Yes, okay, great.
I'm going to mute myself.
Whoops. Wait a minute. I think I'm on the wrong slide here. Let me stop this a second. Stop sharing. Oops. Isn't it the way it always happens? And show.
Well, while
All
John's
right.
loading up, I also want to thank Hannah Luchin for coming and helping with the conference as well. So thank you, Hannah.
There
And
we go.
Matt Healy. Thank you, Matt Healy.
Thank you, Anna and Max. I agree. Okay, we there now? We're at the beginning. I was at the end before.
Great.
Um, the title of the talk, as you read, is Heliconia New Classification and Complete Conservation Assessment. This has indeed taken actually not just 40 years, but 45 years to complete. Heliconia turns out to be a much more complicated genus, uh, both ecologically and evolutionarily than I suspected at the beginning. It also is very widely distributed, as I found out over the course of 40 years. But I think we've made some real progress, and I really thank my three colleagues listed here on this slide: Monica Carlson from Missouri Botanical Garden, Tomas Farr from Charles University in Prague, and Gary Krupnick, also from the same museum that I'm from at the Smithsonian Institution. Between the four of us, we worked very hard over the last probably 5 years or so, analyzing data, putting together conservation assessments so we could present this summary to you today. Um, I also wanted to mention that getting these two papers that I'll describe right now done in time for the conference, one was published in January and the other just a few weeks ago, is also a lead up to a complete monograph of Heliconia. Again, something I've been working on and thinking about for over 40 years. And I think we now have the information in terms of a new classification, species delimitations, conservation assessments that I can put together a complete monograph, which I hope to have done in the next year or so. So let's launch into this. Okay, I'm going to divide this talk into two parts. As you can assume, the first is going to be on phylogenomics and a new classification of Heliconia. as I said, just recently was published, and then we'll go into a conservation assessment of every species in the genus. So to begin with the first part here, this paper, which we entitled Phylogenomics and a New Classification of the Tropical Genus Heliconia, was published in a journal, an international journal called Phytokeys in January. It was an effort between Monica and Tomas in Prague and myself. Uh, what we did essentially is explained on this slide in a very uh, fast way. If you have any questions about the methodology and the sequencing and the analysis, please talk to Monica, who's sitting there in the audience with you. She can give you all the particulars. But we started with 187 species of Heliconia. We took tissue samples uh, and was able to extract DNA from 136 of those species. So about 3/4 of the species are in this analysis. We used 5 outgroups from the Zingiberales to polarize the characters. DNA was extracted from dried leaves and also herbarium specimens. We've made a lot of progress for those of you that aren't familiar with these techniques in the last 20 or 25 years of getting high quality DNA out of often very poor specimens. And Monica is very proficient at this. So we were able to get this DNA out of, as I said, 136 species. And we were able to not just generate loci, uh, DNA loci for a few loci, which is kind of the standard, or was the standard process, but we were actually able to actually generate whole genomes, or at least big parts of the genomes of all of these species from the tissue samples we had. So we had over 450 nuclear loci in the analysis, which is a big data set, to say the least. It didn't answer all of our questions, but it answered a lot of our questions. So we can always add a little bit more in here. So that's kind of the basic process. And what we're trying to do, for those again who aren't familiar with this, is try to understand how species of Heliconia evolved from a common ancestor. That's what a phylogeny is, and that's what we were trying to do. And we can best do that. We used to do it with just morphology. We can now do it with DNA sequence information. And with high quality DNA sequence information, we now can get very good hypothesis on how all these species are related to each other and how they evolved. In this case, probably over about 5 to 10 million years. So what we found that we did have a well-defined phylogeny, and you can see that over here, with almost 100% what's called bootstrap support for most of the major lineages. Uh, that robust evolutionary framework included 5 major clades, 5 major groupings within Heliconia, and 17 minor clades, and I'll describe all those in excessive detail. Even though we did not have all the relationships among these clades resolved, we were able to clearly and strongly identify these 5 major clades and 17 minor clades. Uh, the 51 species that we did not have genomic data from, we then were able to, using morphological features, place them on that phylogeny. So we had all 187 species actually in there. And that's what the classification uh, was built on. So the last point down there, the phylogenetic tree served as the foundation for this new classification of the genus. And here it is. You probably can't even see it very well from where you're sitting. But helicon or phylogenies are now often represented by these circular, what are called cladograms. And here are these 5 outgroup taxa, if you can follow my cursor. And then all the 136 species of Heliconia that were in we had DNA from are represented here. And you can see they're all in kind of um, um, bifurcating, branching series. And that tells us what the general relationships of these 5 major clades are, and then the relationships of all the minor clades, all the way up to the relationships between pairs of species. So it's a very detailed description of the evolution of the genus Heliconia at the species level. Now, it's very hard to see this. So what I've done is I've summarized the phylogeny, which is over on this side of this diagram. And showed how we developed the phylogeny itself, or converted the phylogeny into this new evolutionary classification of the genus. So let me first point out there are these five major branches here on this side. Russ, can people see the cursor that I'm showing here?
Yes, they can.
Okay. So there are five, these are the five major clades, and we've decided to call these five major groupings subgenera, which is a classification, a taxonomic classification, kind of fits the broadest uh, division within a phylogeny. There are five of these: subgenus Heliconiopsis, subgenus Stenoclamus, subgenus Taniostrobus, subgenus Colubrosi, and finally subgenus Heliconia. Each of these five subgenera are then also divided into a number of smaller clades, these 17 minor groupings that I was talking about, and these we have designated as sections, taxonomic sections. And as you can see at Heliconiopsis, there's two sections. Stenoclamis, there's two sections. Taniostrobus, there's two sections here. Colubrosi, there's two sections. And then subgenus Heliconia is the largest with 9 different subgroupings or sections within that. And you can see some divisions here are very well supported as dichotomous branches. Others are not so well supported. So you can see there's 3 branches coming off here. So we haven't resolved those. And here within subgenus Heliconia, there's not all the branches are resolved. So we still have some work to do in terms of figuring out the final evolutionary relationships among these minor clades within the groups. So, and here's the Heliconia representing each of these 17 sections and 5 subgenera on this side. Now, if anybody has any questions during the talk, you can ask me or you can wait till the end. It's up to you. So to get away from all the branches, which sometimes are confusing, I represent the same classification here. I call this a synoptic classification. Uh, and it has the same information, the 5 major clades, 1 subgenus, 2, 3, 4, and 5, and the sections within that, but it's a little less complicated to look at. Let me go over these quickly before I go into each section specifically. So here's that subgenus Heliconiopsis with the 2 sections, section Heliconiopsis and section Perplexi, and I'll explain why we have that name in a minute. The second subgenus, Stenoclamis, with section Stenoclamis and section Angusta, the 2 sections within that subgenus. Here's the third subgenus, Taniostrobus. Here's 1 section within that, section Taniostrobus, section Aurantiaca. Here's the 4th subgenus with section subgenus Colubrosi with section Colubrosi and section Curvy. Notice that within each subgenus, one of the sections has the same name as the subgenus, and that's just the protocol, the taxonomic protocol of how you name things. And then the final large subgenus, subgenus Heliconia, has 9 different sections: section Heliconia, section Lasia, section Conastrum, section Longiflori, section Lania, section Grigzia, section Longi, section Episcopalis, and section Pistazi. Now, remember, we published this whole thing in January of this year. So if you want all the details, it's all in the publication. And I think there must be some copies of that PhytoKeys publication that you can look at if you want. So this is kind of the summary of the synoptic of the classification. And now what I'd like to do, and I hope this isn't too boring, but since you're all lovers of Heliconia, you're going to see a lot of images, but I'm going to go through relatively rapidly each of those 17 sections to give you some idea of the characteristics and the species that are in there. So let's start with subgenus Heliconiopsis. As I said, there are 2 sections within subgenus Heliconiopsis, only 10 species in total. And this is the first section, section Heliconiopsis itself. You can see there's 6 species here. This is that world famous Asian Pacific clade of Heliconias that we knew very little about until about 30 years ago, when a few of us started to explore the South Pacific, all the way from Samoa, all the way to Indonesia and Papua New Guinea, and found out that indeed there were real heliconias in the South Pacific. Little was known about these things. They were all put into a single species for a long time. Uh, but they are certainly good heliconias, but they're very different. And those of you that know the Pacific heliconias know that they're green bracted. They don't have the bright colored bracts. They're green flowered, even though many of the neotropical ones have green flowers. They all have these bright orange or bright red fruits, very different from the neotropical species of heliconia that all have blue fruits. And what's also interesting about these heliconias is some of them, such as Laufau, Solomonensis, Papuana, have nocturnal flowers, flowers that open at night. That's because they're pollinated by bats. All neotropical heliconias have flowers that open during the day and they're pollinated by hummingbirds. Uh, the pollination systems are more complicated than that, and I won't go into it. But these are the 6 species found there. So that's the first section of Heliconia, subgenus Heliconiopsis. And here's the second section, and this is a perplexing section. And we decided to call it section perplexity for that reason. These are 4 neotropical species, primarily Andean species of Heliconia, that no matter how hard we tried to pull them away in the phylogeny from the Pacific Heliconias, we couldn't do it. They are allied with very strong molecular support, genomic support, to be the sister group and a second section in this subgenus Heliconiopsis. They all have diurnal flowers. They have bright red bracts, and in this case, they do have green flowers, but they all have blue fruits like other neotropical heliconias. There's only 4 species of them. They're strong clade, and their sister very close to these Asian species. So perhaps these gave rise to the Asian species, or vice versa. We still have to figure all that out. Okay, let's move on to the next subgenus. This is subgenus Stenoclames. There are 2 sections in this subgenus. This is the first one, section Stenoclames. And all of the species in this section are represented here. There are only 9 species in section Stenoclames. They all have erect inflorescences. They all have resupinate flowers. And a resupinate flower, as you can see here, they're just flowers that stick out of the bracts and they turn upside down. And you can see each one of these has a flower that's outside of the bract and turned upside down, which is a very characteristic of many species of Heliconia, and it's true for all of these species in Stenoclames. All of the flowers are characteristic in this section by being either yellow or orange, some cases greenish, and almost all have this green dot at the tip of the flower, and that unites them together. So that section, Stenoclames and subgenus Stenoclames. The second section is called section Angusti. There are 6 species in this. They also all have erect inflorescences and all have more or less red bracts. The flowers are not quite as fully resupinate in most of the species, as you can see in Velosiana here, Sempowiana, Farinosa. In some, they are resupinate, but they're semi to fully resupinate in these species. As I say, there are 6 species in this group. They're primarily found in northern South America, in French Guiana, Suriname, parts of Brazil, and very distinctive in that way. So that's the second section in subgenus Stenoclames. The third subgenus Taenia strobus. This is the section Taenia strobus. This is a large section of heliconias, and here's just a few representative species. There are 34 species in section Taenia strobus. A few are represented here. This is where we start getting a few species that actually have mostly erect inflorescence, but a few with pendant inflorescences here. And this is an independent origin of a pendant inflorescence from the old world heliconias, the Pacific heliconias, plus some of the new world heliconias also. These flowers are generally not resupinate, and they're enclosed within the bracts, and you can see some of the flowers represented here. You'll probably recognize many of these species. They're found primarily, actually, all over the tropics, Mexico, Central America, South America, quite widely distributed. Again, 34 species in section Taenia strobus. The other section is section Orontiaki, and here there are only 5 species. They're primarily found in tropical Mexico and Central America. They don't really get down into Central America. They have erect inflorescences, but they have some of them, not all of them, but some of them have a unique leaf arrangements, which we call gingiber-like, in which the leaves are parallel up the stem rather than all emanating from the bottom from a pseudostem at the base. Most of the flowers are resupinate in this group as well and held at right angles. Again, only 5 species in this section. Going on to the fourth subgenus, Colubrosea, many of you may recognize some of these things and have heard the name Sigmoidea. For a long time, we were calling these heliconias in this group, and there are quite a few in this group. There are 35 species in section Colubrosea with pendant inflorescences, but they have these very peculiar flowers that are kind of S-shaped, hence Sigmoidea. You can see them here. We decided to actually change that name, and instead of calling this S-shaped as Sigmoidea, we called it snake-like, which is what Colubrosea means because of this snake-like appearance of the flowers. All of these species in section Colubrosea have pendant inflorescences. You can see the bracts can be spirally arranged in some cases or disticus here, but they all have these very distinctive flowers. They are found throughout tropical Central America and South America, very widely distributed as a section. And the second section, we call section Curvy because their flowers are a little different. You can see here, they're not quite that S-shaped. They're more curved, so we call the section itself Curvy. These are a little bit more S-shaped and ally to section Colubrosea. 4 species, they all have erect inflorescences. They all have these primarily disticus bracts, and they're found in primarily Andean South America, not in Central America at all, or not in Amazonian lowland South America. All right, I hope everybody's hanging in there. I know this is a bit grueling, but if you love heliconias, you got to go through it. So let's go on to this 5th subgenus Heliconia. I see Russ there, all okay?
Yeah, no, I was going to say some people are literally hanging on the edge of their seats, so please.
Oh, I hope they are. I know I am. Okay, so we'll go to that last big subgenus Heliconia. And this includes, as I mentioned earlier, 9 sections, very diverse. The first section Heliconia, I'm sure all of you recognize. These have many of our favorite and some of the most popular ornamental heliconias, including Heliconia caribea and Heliconia beehi, and then others with these big, bold disticus bracts, and the flowers sunken down inside the bracts, also somewhat S-shaped, but they have this very long floral tube. And that's what ties all of these heliconias and section heliconia together. They are scattered primarily, well, actually, throughout South America, Central America, and in the Caribbean. There aren't too many heliconias in the Caribbean, particularly the Eastern Caribbean and the Greater Antilles, except in these 2 groups, and primarily Caribea and beehi. I'm sure some of you recognize Wagneriana also, which is very commonly cultivated heliconia. So that's the first of 9 sections in subgenus Heliconia. The second is subgenus Lasia. This is, by the way, all of these in the last section Heliconia and here in Lasia, all of the plants have a mucil-like arrangement of the leaves. None of them are really gingiber-like, and we'll get to that in a little while. But these inflorescences are erect. They all have these resupinate flowers, just like I showed back in some of those other sections in some of the other subgenera. This character of resupinate flowers was often used like an erect versus a pendant inflorescence, which was a very distinctive character and used in early classifications of Heliconia, these racupinate flowers have also been used to designate major sections or subgenera within Heliconia. In fact, Leonard Anderson put all Heliconias with racupinate flowers in his section Stenoclames. But we're now finding that this has evolved numerous times throughout Heliconia, and we find it in a number of sections and a number of independent sections here in Heliconia. There are only 4 species in this section. They're primarily tropical South America. They do not get up into Central America or the Caribbean. Third section, section Canastrum. Many of you are probably familiar with this section. Heliconia section Canastrum has 14 species, and I only picture 5 here, but they have these usually red disticus bracts and these inflorescences that often stick way above the plants themselves. They have flowers that are racupinate, but it's usually with flowers with yellow perianth and some sort of colored ovary. You can see here it's red, red, here it's yellow, here it's green and white. Very distinctive, and many of them have what we call canna-like or zingiber-like arrangement of the leaves. So this habit has evolved also a number of times in Heliconia. This includes such species as Mathiaceae from Central America. I don't show it here, but Heliconia vaginalis, I'm sure many of you are familiar with that species. A lot of different species. I'll say the taxonomy of this section Canastrum, is still a bit difficult. And we're just beginning to recognize some species. Like, I don't have it here, but Heliconia montana, species named by Jose Abelo and Gustavo Morales, I've only just recently recognized as a species, and mostly because of our molecular analysis. So that molecular analysis has tell us something about species designations as well. So Canastrum, a bit complex, more to know about that section. Section Longiflora. I'm sure some of you recognize these species, particularly Heliconia longiflora, which is the name of the section. This section has only these species, only these 6 species, relatively small plants. They all have very distinctive zingiber-like leaves, and that's part of the thing that ties them all together in this section. Flowers are racupinate, as you can see here. Some are quite large, as in Heliconia longiflora itself. Found in both Central and South America, primarily South America. All right, we're getting towards the end of this subgenus Heliconia into section Lania. Section Lania has only these 6 species. Excuse me, let's see, the species that I show here. Yeah, 6 species. I showed 2 different aspects. Some of you may recognize Heliconia lingulata and this thing that we've called pseudoamygdiana, which is actually a different form of lingulata. And so I put it in here just so people would recognize that. And then things like amygdiana and subulata. Actually, the first species of Heliconia I named Heliconia zebrina. But these members of section Lania all have erect inflorescence, primarily disticus bracts, except a few weird things like pseudomygdiana, and flowers that are racupinate and stick outside and are at right angles to the bracts. Tropical South America exclusively, you only find members of section Lania. A few more sections here. Section Grigzia. Many Heliconias had been put in section Grigzia before, but the molecular analysis has now indicated to us that this section only has 3 species in it, 3 legitimate species. And they're all pictured right here. They all have pendant inflorescences, quite large inflorescences. That's why they have the names Heliconia gigantea, Heliconia titanium, and Heliconia grigziana, which named after Griggs, a Heliconia taxonomist. Heliconia titanium is a little difficult in that it's very possible. And if you look at these flowers, you don't see any open flowers. It's very possible that this is a hybrid between Grigziana and another species. And Carla Black pointed that out to me, and I think she may be right about this, but we're investigating that a little bit more. So Grigzia, we get into the pendant Heliconias. This is Heliconia section longi, and this has 32 species in this section, so it's one of the largest sections. All the species are pendant. They primarily have disticus bracts, although there's a lot of variation within here. You can see the flowers all are kind of at right angles. They stick out of the bracts at right angles. Also have a long floral tube. Here's another one. Some are straighter, but very distinctive, all with pendant inflorescences, uh, and none of the flowers are resupinate. Two more sections. Section Episcopalis, again, primarily species with pendant inflorescences, except for Episcopalis, and I'll mention a bit more about that in a minute. They have very distinctive flowers, primarily straight flowers, often with this little curved free sepal here. And the flowers are enclosed in bracts. They're primarily found in Central America and South America, not in the Caribbean. Heliconia episcopalis is interesting that it's one of only two species. First of all, it has an erect inflorescence here in this section that has all the rest are pendant species, but these bracts are deciduous. And I'm sure many of you have collected this in South America, and you find these bracts, they fall off, very similar to Heliconia juruana. And you can see just where the bracts were attached before. So it's a characteristic, and I don't know what the whole ecology of this thing is, but these two species with these bracts that fall off as the inflorescence matures uh, in this one section Episcopalis. Uh, and these are found in both Central America and South America. The final section, whew, got through 17 sections. Here is the section Pistazi. There are only 5 species. Some of you may recognize some of these found in both Central America and South America. They all have pendant inflorescences. Most of the bracts are disticus, except for a little spiral here in Platystachys. Here, Chartaceae, I'm sure this is sexy pink, one of the most flamboyant of heliconias. I put in the green form. This is variety Miana from Venezuela and the northwestern corner of Brazil that has these green bracts, kind of reminiscent of those Pacific heliconias, but not related to it at all. Only 5 species, and there, as I said, both in Central America and South America. Thank you. You all made it through all sections. There's a lot more detail here. If you get the paper that we published, you can find out a lot more about the characteristics of each of these 17 sections and 5 genera. But we could not have done this by any means without the phylogenomic analysis. We had to have that large genomic data set to be able to identify these 17 different sections and 5 different subgenera, and then look for characteristics that united them and begin to understand how these things evolve from each other. Just taking a sip of water here. Okay, so that's part 1, and we're about halfway through. So, yeah. So what I'm going to change gears a little bit because after we got through this whole classification and this new phylogeny of 187 species, we're thinking, well, what are these 187 species doing out there where they're native and what's happening to them and what do we need to know about them? And how are we going to learn more about not only their taxonomy and their evolution, but also their ecology, which I've been interested in a long time. Of course, heliconias grow in habitats such as this one, this cloud forest in Ecuador, but these forests are not static. These forests are suffering some of the the largest alterations of any forest in the world. And I'm not going to dwell on this aspect of it, but we now estimate conservation scientists that at least 1 million species of plants, animals, and fungi are at risk of extinction over the next several decades, up until probably by the year 2050. I know all of you are aware of this. We should all be aware of it. Anybody that goes into the field sees this happening, not just in where heliconias are found in Central America, South America, but all over Asia, Africa, everywhere. It's extremely disappointing, and it points up why we even decided to do a conservation assessment. Why are we getting this destruction? Well, I like to just put one more slide in to remind everybody what's happening here. It's all because of us. It's all because of humans right now. There's been other mass extinctions such as this, where millions of species have gone extinct, but this one's being caused by us. And I just list 8 different primarily causes of this sort of thing, including the degradation and destruction of natural habitats, which I just showed. Pollution, pollution by chemicals, pollution by plastic, of air, sea, and um, land. Exploitation of species, not so much in Heliconia, but many species are collected in the wild and are taken out of that, particularly rare species. Invasive species. We know that invasive species are crowding out Heliconias in some of their natural habitats. We also know that diseases like invasive species are spreading from their home territories into new territories, and that spread of diseases is causing the decline of many species. Urbanization, as areas, particularly places like the Amazon and places like Africa, as cities grow larger and people are expanding urban areas, that is causing the degradation of habitats outside of that. And the two major ones at the end here are climate change. And climate change, it took us 50 years to understand how important climate change was due to the use of excessive use of fossil fuels. But that is a major reason for why many species are declining right now. And a big one that we all could not be more aware of right now is conflict and war. You look at any of the habitats where there are major conflicts, battles, wars in Africa and Ukraine and Gaza, the habitats are absolutely destroyed. And any living thing in those areas, including humans, are having a rough time of it. My explanation, they're just right now on the planet, too many people, not enough resources to take care of all those people, and a vast inequality in the use of those resources. So anyway, all of this together led Monica and Tomas, and particularly Gary, who's a conservation biologist by training and by heart, to say, well, what can we understand about Heliconia and how can we help to conserve these 187 species in all the different forms of Heliconia? So we looked into what was known about conservation assessments of these species, and we found that over the last 50 years, maybe 20 species of Heliconia had IUCN conservation assessments. Certainly not enough to help us understand and to conserve Heliconia. So we took it upon ourselves to go through all 187 species using both our own knowledge and our field experience and our field observations and collections of Heliconia, and all of yours as well. Uh, Plus, we used herbarium specimens that are a permanent record of where species have been collected. And we had over 10,000 specimens of Heliconia in the taxonomic record in herbaria around the world, which we tapped for information. We assigned geographic distributions to all these specimens and species. We assigned elevations, we assigned biomes and ecoregions, and then we evaluated the current efforts in what we're calling both in situ and ex situ efforts to protect these species. In situ being protected habitats and natural areas, and ex situ efforts being growing heliconias in botanic gardens and private collections around the world for ornamental purposes, but also as a conservation measure. And then we performed what's called an IUCN conservation assessment. And I'm not going to go into the details of that. Gary, I think, will cover that in the workshop later this evening. But there's a kind of formula for using all this information from specimens and protected areas and habitats and things to come up with an IUCN criteria of conservation evaluation. What we found, shown on the side here, are that heliconias are native to 42 countries. Maybe some of you didn't know that. But when we plotted them all out around the world, we found that 42 countries account for most of the native species, for all of the native species of Heliconia. Most species of Heliconia are found in 3 countries: Colombia, Ecuador, and Panama. That doesn't mean species not in these 3 countries are not important, but these are the 3 biggest countries. Endemic species, endemic meaning species that are only found in a single country, endemic species of Heliconia are found in 12 countries. And again, Colombia, Ecuador, and Panama account for the most endemic species. So that started giving us an idea of where heliconias are found. uh, and something about their endemism and native habitats. This is kind of the summary, and I'll go into a lot more detail about this in a second. This is the summary of the conservation assessments. And IUCN has listed 5 primary conservation categories: least concerned, near threatened, vulnerable, endangered, and critically endangered species. And when we did our conservation assessments of these 187 species, we found that some of them are doing fine. They're least concerned. They're not threatened by anything. They're widely distributed. We shouldn't be concerned with them right now. Many of them are cultivated, and 86 out of that 187 species are of least concern. 14 species are near threatened. They are they're on that verge. They could go one way or the other, but right now they shouldn't be our priority for conservation. But then these 3 categories, 26 species in the vulnerable category, meaning that they could go one way or the other and mostly are going to become endangered. 38 endangered species, and then 23 critically endangered species. And it's these 3 categories that we're really most concerned about in terms of conservation and protection. You add these 3 categories up, and that's 87 of the 187 species of Heliconia. Almost 50% of the species of the genus Heliconia are threatened with extinction, and that is problematic. There are not too many, usually conservation assessments are done in geographic areas. So you look at all the species that are endangered in this county or this state or maybe this country. Usually we don't do complete conservation assessments of a whole genus like this. And so this is a little bit unique, particularly for a tropical group of heliconias. So a little bit more of detail on those results. First of all, look at a couple of the kind of biogeographic and ecological categories here. I said we looked at mean elevation of all these 187 species, and we looked at the different types of biological biomes where they're found. And for mean elevation, here is lowest elevation going up to the highest elevation. You don't find heliconias above 3,000 meters. So here's coastal and then moving up. And what you see is the majority, most heliconias are lowland. And you all know that. But each of these columns is the conservation assessment of these species. And you find that these lowland, where most of the species are found, these lowland elevations, most of them are not threatened. As you go up in elevation to both, and primarily cloud forests, there are fewer species in cloud forests, but that's where the highest levels of endangerment are, both in terms of critically endangered, endangered, and vulnerable. Oh, I have some arrows here. Okay, there. So where you find the fewest heliconias, most of them are endangered there. If we look over at biomes, and I won't go into this in too much detail, but the whole world is developed or defined by various ecological biomes or ecologies where plants and animals are found. Heliconias are found in 7 of these listed here on this axis. And here again are the conservation assessments on this axis. And almost all heliconias are found in this one biome called tropical and subtropical moist broadleafed forests. And these can occur at a number of different elevations. And because most heliconias are there, also most threatened species are found in this one particular biome. Not so exciting, but it told us a lot about where heliconias grow. Now, what about conservation and protection of these heliconias? This was interesting and a little bit troubling, and this is where we have work to do. Here is in situ conservation, and here is ex situ conservation. And again, here are the number of locations or protected areas in which various heliconias are found. Here again are the conservation assessments. And if we look at this, we find the majority of heliconias found in the most protected habitats are least concern. So we're protecting the species and protected in national parks and nature areas and things are the heliconias least threatened with extinction. And it's the species that are most threatened with extinction that we're not finding protected in any protected areas. So in terms of in situ conservation, we have a lot to do. We need to move these threatened species up here into these protected areas so we can protect them in their natural habitats. We kind of find the same thing when we go over to ex situ conservation, which is what heliconias are preserved in uh, gardens and botanic gardens and private collections and such thing. Again, here's the conservation assessment here, the most critically endangered in this area and the most least concern here. And here are the number of gardens. And what do you see? The heliconias that are in the most number of gardens, the highest number of gardens, 5 or more, are those that are of least concern for protecting. Whereas you go down to those heliconias that are most in need of protection, they're not cultivated anywhere. And even when you look at heliconias that maybe are in one garden or one collection, there still are a lot of endangered species in that. So we have work to do here in terms of getting, again, these species that are most threatened up into these categories of ex situ conservation and the same with in situ conservation. Oh, more arrows. One last thing I want to mention before we open this up for some questions and discussions for those of you that want to do that. We also then thought, and this was one of Monica's ideas, well, why don't we plot these conservation status of all the Heliconia species on our phylogeny? We now know all their evolutionary relationships. Does that have anything to do with the conservation status of Heliconias? I mean, are Heliconias evenly rare and endangered species evenly distributed around the phylogeny, or are they clumped in some way? And when we started mapping these things, we found out, well, yeah, there are certain lineages, certain sections here that have a lot of green here, here, and here, and some that have a lot of red and orange and yellow here, here, and here. And so it looks like the threats to Heliconia are not evenly distributed around the genus. They're clumped in different sections. And so to illustrate this, I go back to that schematic synoptic phylogeny showing those 17 sections in those 5 uh, subgenera. And again, here are I've color coded, or actually Gary did all this. He color coded in these pie charts the conservation status of the species within that subgenus or section. And again, you can see there are some sections, such as Lania, it's almost all green. So this section has very little threatened endangered species. Same with section Stenoclamis, same with section Piscopalis. And you can just compare those with a lot of green or least threatened, those with a lot of orange, red, or yellow, or most threatened. So we look at some of these things. Here's section Colubrisi, not so much green, a lot more red and yellow. Here's section Perplexi with just 4 species, but there's only one that's just barely in there. The rest are threatened. Here's another one, section Longi. So it begins to tell us that there are not just species of Heliconia that are threatened with extinction, are higher threatened, but there are actual lineages within Heliconia, whole groups of species that have the same and have high endangerment and high conservation. I've done this also with the looking at ex situ protection or in situ protection, ex situ protection, and I won't go into this in detail, but we can see there are lineages that are very well protected and not particularly endangered in parks and protected areas. And then there are some lineages that have few uh, protection in protected areas. And the same with botanic gardens. There are some lineages such as Heliconia, and it's in every botanic garden in the world, uh, Episcopalis with rostrata and such things. And there are some lineages, again, like Angusti, like Curvy, like Longi, that have very few of their species protected in an ex situ fashion in botanic gardens. So Gary and Tomas and Monica and I said, well, can we use all this information we just generated on conservation status, on lineages that are higher in terms of endangerment, on those that are in ex situ protection and those that are in situ protection, and come up with some sort of priority list of those 187 species to say, what do we need to focus on most right now? And here it is. And in that paper, this paper on conservation assessment of Heliconia, which just came out, as I said, about a month ago, we have a list in there of 45 priority species, and we give them priority one, priority two, priority three, based upon their conservation assessment, their in situ protection, and their ex situ protection. So we can now, you can hold this list in your hand and say, okay, I'm going to go and get these species as soon as I can in our garden, along with other species of Heliconia. But let's get some of these priority species protected as fast as we can before their habitats are destroyed. So a summary, did a phylogeny, created a classification from that phylogeny. We then went to a conservation assessment of all 187 species. We were able to look at where they're distributed in which biomes, at which elevations. We then were able to also map these conservation status on the phylogeny itself and find out that there are lineages that are in more trouble than others. And then finally, as I showed just in the previous slide, come up with a priority list for which heliconias we're going to concentrate on more. So what Monica and Gary and I and Tomas are asking is, and that's why I wish I was there in the room with all of you and could go to Lion Arboretum and could go to Waimea. So let's all work together to protect as many of these species and lineages as possible before it's too late. And I think we now have the tools, because of this work we've done over the last several decades, to actually prioritize these species and get them into gardens and get them into protected areas as fast as we can. A lot of people to thank. We can go on, but I think I'd rather hear your comments. So I'm going to stop there and open it up for questions and comments if there are any. But thanks very much, and thanks for everyone's help to get these heliconias in protection as soon as we can. Okay, I'll stop sharing the screen so we can.
Thanks so much, John. Can you hear us?
Yes, I can.
Okay, great. So we do have at least one question, and then feel free to either ask your questions and I'll repeat them, come up if you want to see John face to face because he'll be able to see you on camera, or you can also WhatsApp chat them to me and I'll ask them. I'll start with the first question there, John. Can you please describe how to identify or differentiate between Heliconia longa and Heliconia stilesi? And that was from Christian.
No, I can't. Now, yes. First of all, you can tell them geographically. Heliconia longi is primarily on the Atlantic coast, particularly in Costa Rica and Panama, and then down into Colombia. Heliconia stilesii, which is named after Gary Stiles, who was one of the greatest Heliconia ecologists, is found primarily on the Pacific side. It has much more congested bracts, longer inflorescences than longi, and they're primarily disticus when you find them. There's a few floral characteristics that you'd have to measure flowers and find out as well. But they're extremely closely related, and they were all sunk. Stilesii was within longa until I actually saw it in the field and decided that it deserved a different status.
Hmm.
I hope that helps.
Great. And if he has a follow-up, I'll make sure to get it to you. Carla?
Okay.
So regarding in situ conservation designations, once a plant is in 5 conservation areas by waving a magic wand, does that automatically take it out of a most threatened status? So does the evaluation system set up to make plants most threatened will never be in the 5 in the 5 protected areas category?
Yeah, no, that's a really great point, Carla. Though that cutoff of 5 was just because we didn't want to count all the way up to more than that. Gary Krupnick might want to answer that too in terms of IUCN categories, because there are some cutoff points. So some species usually the widespread species are in the most protected areas, and that's what we were trying to show. The rarer the species are, it's less likely they're going to be in more than one protected area. So there's a kind of a relationship there. But I'm not sure if there's just a category that's going to be yes or no. Gary, hi.
Hey there. Hey, John. Thanks a lot. So I'll just emphasize, yes, having plants in protected areas leads to it getting shifted from one category to a next. In the IUCN process, what you have to show is whether or not plants are facing any danger. And having those plants in protected habitats matter. But we also know that protected habitats aren't always fully protected. Protected habitats are undergoing climate change. They're undergoing invasive species issues, and there is still a lot of removal of species in protected areas. And so you have to take all of that into account, knowing what's going on within each habitat. Just because things are so-called protected truly doesn't mean it might just be what's called a paper park. It's just something on paper, but unless there's a real structure to protect those plants, we just don't know what's going on until someone's actually studying it. Next question.
Great, Gary. Yes, let me also say and kind of qualify what we did. Our data set, even though we used 10,000 records of these things and are also our own observations, many of these species are still poorly known. We only know them from 1 or 2 or 3 collections. And so there's a lot more field work needs to be done. But this was an attempt to say, this is what we know today, 2025. This is how we assess what their conservation status. My feeling is it's not going to get any better for most of these species. It's going to become more threatened.
Thanks, John. Thanks, Gary. Any other questions? We got Rob.
What's happened with Heliconia longissima? Is that being merged into Excelsa or the other side?
So the question was from Rob, what's going on with Heliconia longissima? Is it being merged with Heliconia excelsa or longa?
Yes, it is being merged. And that is what the monograph is going to tell you. We don't have all that information out there right now. I was going to put that, or we were going to put that into the classification paper, but it's just too much work to do, and that's what your monograph is about. So I'll have synonymy listed for all those things. Right now, longissima is going to go into Excelsa, as far as I can see. So, and I know everybody may have a favorite Heliconia and say, why isn't that on my list and all this? Well, I've been trying to kind of assess things both in terms of morphology, in terms of distribution, in terms of our molecular results. For example, Heliconia deflexa, I sunk that into vaginalis a long time ago, and our molecular data show that it's a very distinctive thing. And I can't, you have to kind of balance these data or the evidence you have for these things. So that's when the monograph does come out, you'll be able to see all that, and hopefully it'll be soon. And that will change as time goes on, as we learn more about these species. But that'll be the kind of the best estimate right now for me.
Thanks, Rob, for that question.
Do have a follow-up?
Oh, okay. Yeah, of course.
I was told a couple of months ago that Borgiana is now been merged into Campania, I don't know how to pronounce that. Campania. Is that possible?
So Rob was questioning, and he heard a little rumor that Borgiana is potentially merging into Champiana. Is that true?
Quash that rumor. There is no such thing happening. No, those are pretty distinctive. And I've collected both of them in the field and compared them. You go to a botanic garden at Lyon. I mean, you can see it clearly that they are very different species. There is a species that was named in Mexico called Ushtapensis. And I'm afraid that is just, I did sink that into Borgiana because it was a very local variant, but it's not the same as Champniana at all. So those two will remain distinct as far as I can see right now.
Great.
Yeah.
Any more questions? We got time for about one more. Dr. Criley.
Hold on, John.
Okay.
Great. Yes. And then just shake your head if I ask it wrong. So what are the number of genomic sites, John, that you are using to distinguish species?
Oh my gosh. I cannot answer that off the top of my head. Uh,
I'm gonna answer in an email.
yeah, no, but maybe Monica has some idea of how many. I mean, she did that analysis. As I said, we had over 450 uh, nuclear loci that we use, but Monica may have some idea on that.
Hi, everybody. So we started with 980 nuclear genes. We deleted all the ones that were not variable, and we ended up with 452 genes that gave us variation. Among species, like if you're trying to different, like we focus a lot in the Caribea behind, they ended up being sister species. And between those two, there are at least 300 different loci that show variation between those two sister species. So it's quite a powerful tool, and it's available for all Zingiberellus. So the same kit that we use for those nuclear genes, we used it also for the order Zingiberellus, and now some people have been using it for the redescription or redistribution of all the Zingiberaceae genera, like Alpinia is being chopped up in a lot of different things. So this genomic size data set can be used across the entire order with a lot of success. So, I just wanted to say, like, both of the papers that we publish are open access. So you don't have to pay anything. You can just download the PDF from their journals. So anywhere in the world you want to access them, they are available. All our PDF are open access.
Monica, thank you. Yeah, let me just build on that a little bit. We were not really focusing on species relationships at pairs of species in this analysis. We really wanted to get an overall broad picture of relationships and to see if we could define these major clades to serve as a classification. Now it's up to all of you out there and us to continue to focus on various lineages, maybe this section or that section, to see if we could tell more about the relationships among species within those sections. And that's the challenge ahead.
Great. Thank you so much, John, Gary, and Monica for answering those questions. I know from a production side, just outside of the importance for conserving Heliconia species, we want to keep these species around so that we have breeding material, right? From a production perspective, we want these plants to stay conserved. So for you guys, it's very important. So unfortunately, John isn't here, and you may not be able to come for the workshop. Correct, John, because it'll be about 10 p.m. for you. We'll have you loaded up. If you want to come, please do. But Gary and Monica are going to be leading the workshop. We have a questionnaire that we put into each of your swag bags. Read it, and if it's for you, try filling it out early so that you're prepared for the workshop, or bring it to the workshop and we can work on it. I'll give some more information at the end of the day about getting to the workshop, a little bit of logistics, but we'll have that workshop available for you this afternoon. John, just
Can I just say one thing about the workshop, please? Am I still on? Yeah.
Yes, you are.
Again, I really regret that I can't be there, but I'm so pleased that Monica and Gary are, and they're going to run it. I'm not going to be able to get there. I think it'd be too complicated for me to try to participate. But this really is a critical chance for us all to be in the room, or those that can be in the room, to see if we can come up with some sort of strategy and plan to get as many of these things, many of these species in particularly cultivation as we can. Protected areas also, but to come up with some strategy of how we can all work together to make this happen. So I would urge all of you that can be there to go to the workshop, and good luck.
Welcome back. For our next speaker, we've got an individual who has Heliconia in his blood, Dr. Colton Collins. So Dr. Collins was raised on his family's Heliconia and ginger farm in Hilo, Hawaii. where he worked fulfilling orders and caring for their living collection. He's been managing the farm since 2011. He graduated from the University of Hawaii at Hilo with a master's degree in tropical conservation biology, where he researched large-scale diversity and endemism in patterns in Heliconiaceae. He completed a PhD at Nanyang University, Technological University in Singapore, where his research focused on plant and soil interactions in Bornean rainforests. If you want more information, we've got a little bit longer bios in the speaker abstract and bio. So please welcome Dr. Colton Collins.
Hello, everyone. Thank you for coming. Today, I will be speaking about the introduction of gingerbread to Hawaii, and for the twist of it, I'll be speaking about just my family's role in these introductions. I'll be discussing 4 different chapters.
Okay.
Each chapter kind of represents 3 different generations of introduction, and the final chapter will discuss conservation and challenges in introducing gingerbread to Hawaii.
Okay, good.
Okay, chapter 1, we start from the beginning with the distant ancestors, the first Polynesians. They were the first to introduce gingerbread to Hawaii. But before we get exactly into what they introduced, we kind of need to situate ourselves geographically. So right now, you're in the middle of the largest ocean on Earth. It's the most remote island chain. You're about 2,500 miles from Tahiti, 2,300 miles from California, and 3,800 miles from Japan. Any plants here that are native to Hawaii got here by wind, ocean currents, or migratory birds. There are about 1,400 native taxa, and this includes native species, subspecies, and varieties. So some species got here and then eventually evolved, and that's how we got the 1,400 taxa. And 90% of them are endemic. This means there are no native gingers or endemic gingers, heliconias, or bananas at all to Hawaii. So everything you see here, whether it's the bananas you're eating, the heliconias in the landscaping, or gingers in the floral arrangements, those were all introduced.
Here you go.
Thank you.
Sorry.
Okay, so the first Hawaiians got here. They sailed on these double-hulled canoes, and with them they brought canoe plants, or plants that were on the canoes with them. They first arrived in Kauai and then eventually made their way down to Big Island, and this just is a painting by Herb Kane representing the first arrival by these Polynesians to Big Island. To get an idea of what plants they brought, we need to kind of understand where they came from. So it's believed they originally actually migrated from Taiwan, made their way through Southeast Asia, inhabiting the Melanesian Islands. They eventually landed in Samoa, then to the Society Islands, which is modern-day Tahiti. And from there, they branched out to New Zealand, Papua New Guinea, and up north Hawaii, and that creates the Polynesian Triangle. So you can tell by coming from Southeast Asia what plants they could possibly be bringing on their canoes, as well as what plants are native to the Melanesian and Polynesian Islands. And interestingly enough, the sweet potato is actually, there's linguistic and genetic evidence that it was exchanged with Latin America about 1200 AD. So there's even some sailing and exchange going on back then. So these are collectively termed the canoe plants. So these are the 24 plants that were brought with them on canoes. Some argue 26, other 22, but most people kind of settle on 24. This includes your coconuts, your taro, your bamboo, your sugarcane. And if you wanted to, after the zombie apocalypse, restart society, these are the 24 plants you need to restart society on a tropical island somewhere. Included in these 24 are 3 gingerbread. So that's about 1/8 or 12.5% of all canoe plants. So these plants were very important to the survival of the first Polynesians that arrived here. One, like Elena, is medicinal for inflammatory properties. It's also used for dyeing. They wanted to look good too in their clothing. Awapuhi, or shampoo ginger, it's antibacterial. It's good for their hygiene. It's also used to kind of soothe coughs and fevers. And banana, there's a few banana species, varieties that were brought over. And it's arguable that that's the most important plant that they could have brought over, just for its edible, medicinal, and just overall utilitarian purposes. One such purpose that I'm just going to use an example here is the Native Hawaiians wore these feathered capes. And the Ali'i wore the capes to kind of show off their status and their royalty. And they're made out of feathers collected from the different native birds here. But how do you catch these birds, right? So what the Hawaiians did was they made an adhesive out of banana sap. They would put those on branches. The birds would land on the branches. They'd pick a couple feathers out, scrape off the sap, and then release the birds so it could regrow more feathers. So now these capes need tens of thousands of feathers. These long ones need up to 60,000 feathers in order to make. So you can imagine it takes a lot of time and effort to make these plants, make these capes, and impatient people probably wouldn't be able to handle it. So they had a saying for impatient people, and it was, bananas do not fruit in a single day. So this kind of shows the idea of not only the utilitarian of the plant, but also how it kind of shapes their way of thinking and the language as well. And well, if your significant other is rushing you to get ready, or your boss is telling you, hey, where's the report? Just let them know bananas do not fruit in a single day. So from this chapter, the main idea was that the Polynesians were the first to introduce gingerbreads to Hawaii, and that they were necessary for their survival and way of life. Okay, in this chapter, I'm not going to talk about my father, Mark Collins, and his introductions, and I'd like to argue that he possibly introduced the most types of gingerbreads to Hawaii. For some of you, it's a trip down memory lane. For others, Mark Collins was a longtime HSI member. He was president for 2000-2002. He traveled to 50 countries in Latin America, Southeast Asia, West Africa, collecting different plants. I collected, named, and introduced, to my count, over 200 gingerbrelis. He has a couple species named after him, Gingerbread Clinci, Bosenbergia Clinci, and he had a few companies, Eden Farms, Jungle Jewels, and Plant Group. So if you ever receive material from there, it's from his collection. So how did I figure out this number? Well, I used the Heliconia checklist and register, which is a great resource. It names the different cultivars or varieties. I kind of use those terms interchangeably. And it names the year, the nominant, and it also tells you the correct spelling or whether the name was rejected or not. So I went through the registry, found whatever names were introduced by him or through one of his companies. And this is my count. So I came up with 40 different pendant varieties, 92 different uprights, and it's represented by 61 different species and 8 different hybrids. However, he passed away in 2011, and the article was posted in 2005. So between that time period, I don't really have the information on that. So I believe there are possibly more cultivars that are not accounted for in this count. One of my upcoming projects, some of you know that I do a Heliconia poster. I'm now trying to do a Heliconia poster just of the varieties he introduced. I just thought it was a fun way to kind of show his accumulation of work over time. So coming soon. In addition to a Heliconia registry, there's also a Costas registry online that you can check out. So here's just a list of some of the Costas that he has registered. In addition to these, there are several others. I believe there's a couple dozen. Some of them were on the registry, some have not been registered yet, and I have been working on uploading some of his, or registering some of his introductions. When it comes to other genera, we don't have registries. For example, Globa, no one's really keeping track of that. These ones he did have patents on, and he was actually receiving royalty checks for a few $1,000s. But when I took over, they're only for like 50 cents. So I don't know how he got royalties on these plants in the first place, but it was a way to keep track of what he introduced and what he didn't. For others, you have to look in the primary literature. So Greenbells, for example, if you look up the description in the peer-reviewed journal, they do cite my dad as being the one that collected it, and the description was made out of his collection. And other species or other introductions, I did more of interviews with people that collected with him, whether it's wife Bee or Sean Callahan. Also, Tim Chapman, he has a great website. On his website, it kind of shows some of he tells the stories a bit of the introductions of different varieties, and he credits my dad to a lot of those. In addition to the Costas, Heliconia, and Globas, he's also introduced Marantacea, Alpinias, Gingerburs, and some novelty ginger. I don't have those all listed or showed here, but at the end of the day, my count is around 200. It's probably more. The more people I talk to, the more I'm learning about what he introduced. So the main takeaway from this chapter is my father likely introduced some of the most ginger ales to Hawaii. All right, so this chapter now I'm talking about myself a little bit and some of the latest introductions I'm bringing into the islands. So what I like to do is I go to the different markets abroad, exchange with different gardens and botanical institutions, as well as collect things from the wild. And here's some of the introductions I've brought in recently. This Globa hybrid, the Jasmine and Royal Purple, that I just brought in last week and it's actually available at the silent auction last night. So someone was fortunate enough to get those. The Purple Pakia, brought that in a couple years ago. The species mood, actually, I got that from John Mood. I really had to talk him into it because he didn't really want to give it away to anyone. But the bulbils are like the size of a golf ball, and I just couldn't resist. And he eventually gave me one. And a few years later, I was able to grow it and then release it to other growers in the island and the US. This Grixiana Hunter's Moon, that's actually a seedling variation that I found growing roadside on the way to volcanoes. So those going to a volcano, it's just a giant club. You'll definitely pass by it on your way there. And this curcuma was just described a year and a half, 2 years ago out of Thailand. And I was able to bring that in. And there's a number of growers around the islands growing that now as well. All right, these are things I have here in the greenhouse, but are not ready to be introduced yet. I'm building up the numbers before I eventually release these. So wild collecting in northern Thailand, this is one of my favorites, the Zingiber currii with the nice pink tips there. Globa racemosa, that's been a hard one for me to multiply. That one definitely prefers the cooler weather. So that's been a struggle for me to get more of that. This Hydicium hybrid found growing on a cliffside. It's the only kind of patch I've seen in the whole area. So I'm excited. If I tried bringing that in once, it didn't make it, but I brought it in again, and it's growing well this time. And this custis, it's disgustingly beautiful, I like to say, because the nice lavender flowers is quite gentle and soft, but then the inflorescence itself is just gross. But I'm sure someone would be interested in it. I have not been collecting in Myanmar. They're currently under a civil war with like 25 different armed groups, all fighting for power in the past few years. But I know a guy who knows a guy, and I've been able to get some of the ginger varieties over here to Hawaii. Most of them are undescribed species, are currently being described right now, and it comprises of curcumas, globas, and zingibers. I do think this zingiber here does have a cut flower potential as well as potential in the landscape industry with the nice silver leaves. And the past couple of years, I've really been enjoying collecting in northern Borneo, especially in Malaysia. It's very excited to find this Zingiber incomptum. If you look at the stem, it just grows along the forest floor, like Heliconia rectans, how that kind of just grows along the forest floor. This one does as well, and it's like 1.5 feet, 2 feet long. So it's quite surprising for me, and it's quite beautiful with the recurved bracts there, as you can see on the inflorescence. This gingerbread in the lower corner here also has cut flower potential, produces these long inflorescence. It's green, eventually turns red. Some globas, there's globas everywhere, but I like collecting from Borneo because those globas don't go dormant. So that might be more appealing to the average grower who struggles with plants that go dormant and plant it over and think they killed it, that type of thing. And this Plagiostachys is another just weird and gross one, but again, I'm sure someone might find it appealing. To continue on with the Borneo expedition, I just came back from Sabah last week, and I was very excited to find this Hydicium. It's an epiphyte. There was 2 Hydiciums known to the area, Muluensis and Cylindrica, and this doesn't look like either of them, so it could be something new, especially with its thick, deep red bracts. I did not see flowers at the time, just one kind of clump was at this stage. The seeds weren't viable either, but I did collect rhizomes and I was able to bring that back in. There's a bunch of Marantaceae there, but to be honest, they're not very attractive for cut flower or landscape purposes either. And conservation-wise, I think there's just a lot of them. So the only one I did find attractive was this kind of bright orange one, which I brought in. I brought in Hornstedia and a mobum as well. So takeaway from this chapter is that I am continuing kind of this family legacy of bringing in plant material to Hawaii. And now we'll talk about conservation and challenges of introducing gingiberries to Hawaii. So these are species we're currently conserving in the collection. It ranges from endangered to near threatened. This Fernandezii was on the Heliconia priority list that John just talked about. And we've even been able to release those to other growers in Hawaii and in Florida as well, just a couple of weeks ago. So it does show the role of these kind of smaller private collections that they can play in conserving these species. Not just heliconias, but we also conserve a number of ginger species from alpinia to zingiber, globus, and curcumas. It's a lot of work just to get all that information, I'm sure, just to assess the heliconias. But imagine gingers, there's 87 heliconia species, and gingers is 3,000 species. So there's a lot of work to be done. And here's just a handful that we are conserving. It's not a complete list, but just to show you the diversity that we do have in the collection. These were species we're conserving, but lost over time. I like to say it wasn't my fault, but a lot of these were. We had pigs that got in, some we didn't maintain properly, and other plants kind of took over. The Gloriosa, I divided that too much, and it eventually died down. And I just saw this on the priority list too, so that's kind of a tough lesson to learn, but now we know for next time. But it also shows the importance of exchanging material with other growers and other gardens, just so they have a backup repository. And just to note, I did lose Heliconia villosa, and then haven't seen it for a few years. Then on a Facebook group, someone posted a photo of it and asked, "Oh, does anyone know what this is?" There's a grower on Kauai. She happened to buy it a few years ago from me, and it just didn't flower. And then I was like, "Oh, wait, that came from us." So then now we're going to exchange later, so we'll eventually get it back. So that just shows, again, the proof of concept of having these other individual growers growing these plants. Some challenges of introducing gingiberries to Hawaii. In the 12th century, it was saltwater and time. You can imagine crossing the ocean for an undetermined amount of time, and you have to keep them dry or away from all that salt water. In the 80s and 2000s, my dad faced a number of lawsuits trying to introduce Heliconia to Hawaii. There's an established banana industry here, and they're worried by introducing Heliconias that it would bring diseases that would eventually impact the banana industry. But eventually they were able to show in court that bananas and heliconias aren't related, so then he was able to bring in heliconias. And modern day, some of the struggles are the USDA, they're there to protect Hawaii and US agriculture, and they do a good job of that, but sometimes they can seem a little inconsistent of what they allow in and what they don't. Even though you follow all the guidelines on import permit stuff, it kind of really seems up to the individual inspector on what they deem is too dangerous or safe to bring in. And it's quite sad. Just this past trip, I brought in some 16 different Heliconia species that weren't grown in Hawaii from Nanook Botanical, and they all got confiscated and burned. The gingers from the same garden, from the same collection, were allowed in, but for some reason the Heliconias weren't. So just even within the same box, it's just trying to navigate what they allow in and what they don't, and what they deem as clean and whatnot. The other struggle is public perspective. Nowadays, there is a push to grow more native species, and with the number of endemic and endangered species in Hawaii, like, it is good to support the native species. But on the flip side of that, it kind of vilifies the introduced species, especially gingers. Now, there are a number of invasive gingers, which you will hear about later, but not all gingers and gingerberries are invasive. So that's something that the public could use perhaps education or outreach on. And just from a Hawaiian perspective of why we should conserve gingerberries in Hawaii, the first is the Hawaiian value of malama aina, which is the idea of you take care of the land and the land will take care of you. Much like some cultures, they emphasize perhaps education, or other cultures emphasize the close family union. In Hawaii, we emphasize taking care of the land. You can imagine being stuck in the middle of the ocean with a limited amount of resources. Your life really did depend on that land. And so over time, that kind of value stayed with us over time. So I just think within that value, we should be conserving not only Hawaii, but preserving other plants around the world. But specifically, we should conserve gingerbrelis because they did help our distant ancestors survive. And nowadays, they also help local families, whether they're florists or farmers or landscapers. These plants do play a big role commercially and on individual homes and livelihoods. So I think we could return the favor. They've been good to us, so we could be good to them and take care of them. And with that, that's my presentation introducing Gingerbread Hawaii, a family affair. Distant family was the first to introduce. My father probably introduced the most. I'm introducing the latest. And my niece, Kailea, she's showing some interest. She likes the flowers and to make TikTok videos, run through the greenhouse with the sprinklers on. Cleaning rhizomes and pulling weeds is acquired taste she didn't acquire yet, but we have some hope. And with that, any questions? Okay.
Curious, on your Fernandezii, is there any indication that it is subject to elevation where it grows better, not at high elevation and performs better at low elevation? Do you know anything about? Because I know in Puerto Rico, they have farms that are high elevation, heliconias that prosper. And when we try to grow them on Maui at high elevation, they don't do as well. So do you know of any of the reasons why that is?
Okay, so the question was for Heliconia Fernandezii, do I have any experience of why it grows well at high elevation at some points and not at other?
Not at
high elevation, but at low elevation.
Correct.
Well, every species kind of has their own niche that they thrive in. We've tried to replicate what is found naturally. So we have seen it growing at high elevation, but at low elevation, we try to replicate those conditions by perhaps growing it more in the shade where the daily average temperature is lower, or you water it during the hottest days to kind of keep that temperature down. And so if you are trying to grow a mid-elevation or high elevation species, that's kind of the trick we use, but that only works so far. Some species just grow higher or lower. I'm sure you should be able to grow it in Maui because Maui's elevation isn't quite that high enough to. Yeah. Some of these do take, like the Xanthoplosa, it took us like 8 or 9 times of different rhizomes to get established, but once it gets established, it's pretty set. So it might be one of those things where you just have to keep trying, and then eventually you'll find that kind of sweet spot on your place. Yeah. Oh, yes. Monica.
So is it easier to introduce rhizomes or seeds?
The question was, is it easier to introduce rhizomes or seeds? It's seeds. There's less paperwork and inspection that needs to happen for that because seeds are easier to clean and they're likely to introduce less pathogens. Rhizomes are quicker to grow and easier to get established, but they have a lot of crevices that could hide soil pathogens or little insects and whatnot. So even for the permitting process, you can bring in small amounts of seeds without permits, just with phytos, whereas large amount of rhizomes, you definitely need to get that inspected. So seeds is always easier. Oh, yes.
Have you ever received or have resources for tissue culture at Heliconia? I know it's renownedly hard to tissue culture.
Have I received?
Have you done tissue culture in any Heliconia to bring them in? Using tissue culture.
No, I haven't. But with bananas, you're not really allowed to bring any bananas in except for with tissue culture. That's what they allow in.
I'm just not sure if anybody's doing it commercially at least.
They grow fast enough that you might not need it. Yeah. All right. Well, thank you for your time. And if you have any other questions, I'll be here all week. All right. Thanks.
Next up, we've got Dave Lawrence. Dr. David Lawrence is Senior Research Botanist at the National Tropical Botanical Gardens on Kauai. He serves on the Board of Directors for the Heliconia Society International and curates NTBG's living collections of Zingiberales and Rubiaceae. And you can find his full bio in our abstracts and bios. And I'll give you those there. Presentation. Yeah.
Well, aloha, everyone. It's a pleasure to be here. I've been attending HSI conferences for quite a few years now, actually, not everyone, but many of them. As a botanical garden, as Russ mentioned, National Tropical Botanical Garden has introduced a lot of plant species over the years, including some Zingiberales. My own specialty is actually Rubiaceae, the coffee family, gardenias, quinine. I'm a plant taxonomist by training, but I do oversee our Zingiberales collection there. And okay.
Just to arrow over.
So I'll be talking a little bit about some of the dark side of Rubiaceae. I mean the Zingiberales. I've got Rubiaceae on the mind. Some Zingiberales are a little too successful in certain areas, including islands like the Hawaiian Islands, and they can get out of control and cause some real issues with the native vegetation. The order Zingiberales has 8 different families, some 2,600 species in maybe 68 genera, and many of these families do have species that can become naturalized, except for the Loeaceae, it's a small family. So I'll be looking at some of these on a case-by-case basis, and let's focus on the Hawaiian Islands. As we've seen in some of our talks, many Zingiberales are really endangered. The habitat destruction is taking place, many other threats. But, and actually, we have a number of families that actually become weedy, though, especially on islands. Oceanic islands like the Hawaiian Islands have a flora that's evolved over millions of years, sort of in isolation. And when we, it's a little bit susceptible to invasion by other species from continental areas, especially. They evolved in the absence of predators and diseases. So they really weren't, many of the native flora here, it's just not very resistant to invasion by introduced species. And then you've got introduced birds such as bulbuls and other things, minor birds, they spread seed around as well. So in the Hawaiian Islands, there are about 9 different naturalized Zingiberales that have been documented here, including some really aggressive weeds, Heliconia and Chilocostus, which I'll be talking about a little more in detail. So I think it's in NTBG, where I work, is one of the conservation centers for Zingiberales, as is Waimea Arboretum and Lion. And when we were walking through Lion Arboretum yesterday, we saw some of these Zingiberales can get out of control and spread beyond where they were originally planted. So I think as growers and conservation centers, we should keep an eye on these things and try not to let them become naturalized so that they negatively impact our ecosystems. And this could include removal of spontaneous seedlings that you see spreading around your property or into adjacent native vegetation. I know it's difficult to do, but it's kind of our responsibility. So let's take a look at some of the invaders here. One of our bulletin articles, the genus Heliconia was highlighted. It's native to Northeast India and the Himalayas, and Anita Gowda and her student Ajis Ashokan published a couple of nice articles on Heliconia. And it's a favorite plant here in the Hawaiian Islands. You make leis out of them, garlands, They're very fragrant, the Heliconia species. And, you know, one of these leis can go for $50 or $60 each. It takes a lot of time and effort to sort of make the lei. So they're used in, you know, weddings and other special occasions. And they usually use a white ginger here, Heliconia coronarium, which you can see on the map is actually, it's been widely grown. It's also widely naturalized, usually by rhizome cloning. You can find them growing in dense clumps, and also birds spread the seeds. Seeds are red and kind of have a nice arrow that birds, mainly introduced birds here in Hawaii, tend to eat and spread around. The yellow ginger, Heliconia flavescens, is also widely naturalized in different parts of the world. You can see on the map. It's become a major pest in the Hawaiian Islands, and also spreads by rhizome clumping and seeds. There are a few other Heliconia that are growing here, but they really haven't taken off and spread. This one, the red butterfly ginger Heliconia greenii, is cultivated, but just produces plantlets or on the inflorescences. It's not been seen to seed in the Hawaiian Islands. But by far the one of the worst weeds is the Heliconia gardnerianum, the Himalayan ginger, or sometimes called kahili ginger here in Hawaii. This is native to the Indian subcontinent, Bhutan, Nepal, and northern India, and it was introduced to Hawaii not that many, less than a century ago. Now it's become naturalized in at least 4 of the main islands, and you'll see it when you go to the Big Island up in the Volcanoes National Park area and many other upland areas there. It's really produces prolific seeds and dense rhizome clumping too. In fact, on Kauai, it's become the major weed in the upland native forest area. It even grows in the birds will drop swoop seeds out in the mossy branches, and it grows in an epiphyte sometimes, as well as on the ground. And it continues to spread there, now covering over 50,000 acres, I believe, on 5 of our islands. Smothers young plants, young native seedlings, and it's just causing some really serious ecosystem disruptions. This is just a map of the Heliconia swamp area, which is a wilderness area in upland Kauai, taken back in 2006, to showing some of the distribution. At the time, they were taking data points, and now it's actually gotten much worse, spreading, and really no end in sight. It can be removed manually, of course, by pulling it out, but this is very, intense process, and it's costly as well as being labor intensive. They've already spent over $1 million trying to remove in the state here the Heliconia gardnerianum. A few sort of novel methods have been thought of to try to, for example, shoot herbicide from paint, using paint guns from a helicopter. They've tried this on Manoa, which is another invasive in the Hawaiian Islands. Doesn't work very well, apparently, and plus the herbicide is, can be released and kill other plants and move through the groundwater, so that's not a good option. Some other biological control methods have been studied also. And this is probably the best way, or the only really feasible way of controlling some of these gingers, like Heliconia gardnerianum. So we'll take a look at a couple of them that were kind of studied by this CAVI.org, Center for Agricultural and Biosciences International. It was a nonprofit, and it's a collaboration between New Zealand and the Hawaiian Islands, the Nature Conservancy. So they went to the native range of the ginger and brought back some of the insects and other potential biocontrol methods that would, attack the ginger, feed on it. Now, one of which was weevils. There's a couple of different weevil genera, but they have a broader host range, and they can also attack other gingiberales that are in cultivation here. Now, they looked at moth larvae, a genus moths, which was potential also, but apparently not that effective. And then also a fly larva that bores into the stem. So they're, continue to study them further, but it doesn't look like it's going to be very effective controlling it on a large scale. This is just showing some of the weevil damage on some of the stems and their experimental areas. New Zealand also has a real serious problem with this Heliconia gardnerianum. Rust fungus, they looked at some Puccinia fungi that affected the ginger, but it unfortunately also attacks other gingiberales, so that's not going to work either, apparently. So additional field work and research is needed actually for the more biocontrol agents that might be in northeastern India and Sikkim. This little map here just shows the potential for further spread of Heliconia gardnerianum. These would be actually areas that would support the species if it were to naturalize and escape in additional areas. So it's a lot of the southern United States up to the west coast, including Puerto Rico and the Hawaiian Islands. So serious problem. Another family we're going to look at here is the Costacea, Dave Skinner's favorite family. And this is Chilocostus, the crepe ginger, Chilocostus speciosus, also known as Helenia speciosa. It's another serious invader, prolific seeder, produces a lot of seeds, which can be spread by birds. And also many Costacea just spread through canes vegetatively. If they fall over, they'll produce new plants from the nodes. And this is kind of the current distribution of the Chilocostus speciosus. So it's naturalized in a number of different areas. And here's an example of, what the canes look like when they fall over, sprouting up new plants. A couple of other Costacea, Costus woodsonii, which is naturalized in some areas. It's a neotropical species, but it's invasive in Hawaii and the Cook Islands, as well as the Marquesas Islands, a few other islands in Micronesia. Spreads by seeds, canes, and plantlets produced on the inflorescence. Costus pulverinatus, this is another one, which has become naturalized in Samoa, for example, and on Oahu. I think we may have seen one or two of these yesterday that had been spreading in line arboretum. There are a couple of African species, the Costus dubius and Costus afer, which are also pesky. The Costus dubius is naturalized on Oahu and Kauai in the lowlands. It's one of the worst Costus actually in our botanical garden. It's just been spreading all over, and it's hard for the gardeners, the horticulturists, to really tell if this is a weedy species or not. So we have to, work with them and try to eradicate these clumps before they get too big and spread further. What else? Other gingiberales naturalized in the Hawaiian Islands. There are a couple of alpinias, Zerumbit, mutica, and purpurata. Heliconia is not really a problem. I think there are one or two species that are will spread by seed and rhizomes. Canna, the Indian shot, it's naturalized, but it's not really an issue, not a serious problem. And Zingiber zorumbit, which the Avapuhi ginger, which Colin mentioned, naturalized on some of the wet valleys. Your two naturalized Heliconias, latispatha and Bihai, mainly by birds which spread the fruit. Canna, you've probably seen that. It kind of grows along streams often and other moist areas, spreads by seed, but it's not a serious pest at all in terms of weediness. And this is the shampoo ginger or Zingiber zorumbit that was introduced. It's a Polynesian introduction and it was used as a medicinal plant. Doesn't produce seed. It's apparently a sterile species, but it does produce dense, rhizome branching. And it's mainly confined to valley bottoms on the windward side of the islands, some of the main islands. And you can see the dense rhizomes in this lower photo here. Goes dormant in the winter. You don't even know it's there practically until the summer when it starts sprouting up again and flowering. Turmeric, Curcuma longa, is also a Polynesian introduction, a medicinal plant, dye plant, as Colin mentioned. And again, it doesn't produce seed. It just spreads vegetatively by rhizomes, not at all invasive. In fact, it's quite hard to find in the wild these days. We've seen the shell ginger here, Alpinia zorumbit, naturalized in a few areas on 4 of the islands. And it spreads by seed and then also rhizomes. A couple of other Alpinias, Alpinia purpurata and Alpinia mutica, also have been recorded as naturalized on several of the islands. Marantaceae, there are a number of cultivated Marantaceae, but about the only one that shows a sign of spreading is the rattlesnake, Calthea protolifera, rattlesnake plant, sparingly naturalized on Oahu and the Big Island, also in Florida, apparently. It's native to the neotropics. At NTBG, we introduced this Donax caniformis from the Micronesian area, Palau, and actually that one's sort of showing signs of escaping, and it's popping up along our stream, which runs through the Lawai Valley where our garden is located. So we're keeping an eye on this one. It's potentially invasive. There aren't any bananas that are naturalized in the Hawaiian Islands. So when we had the conference in Costa Rica, I noticed this one here, Musa velutina, naturalized up at higher elevations at San Ramon in Costa Rica. And Carla, you probably remember this one. No, you didn't see it there? And along with the beehive ginger, Zingiber spectabilis. I was kind of surprised to see it up there in the native forest. The traveler's tree, Ravenala madagascariensis, it's not known to be naturalized in Hawaii, but I was a Peace Corps volunteer on Mauritius for a number of years, and it's really a serious pest there. It's just covers many hillsides and valleys, spread by seeds, and then it forms dense clumps. It's hard to really move through patches of this Ravenala. So something to keep an eye out here for in Hawaii. I know it's cultivated quite a bit. It does have these blue aerolate seeds that could be spread by birds. And the only family in the order Zingiberales that is not weedy is this Orchidantha, which known from cultivation in a few botanical gardens. It has flowers that are held or produced low to the ground, and they're usually beetle or fly pollinated, so they're not very fragrant or don't have an appealing fragrance, and it's not a real showy plant either. So there are a number of resources that are available for finding out more about invasive species. The Hawaii Invasive Species Council, for HISC. A couple other ones, Pacific Island Ecosystems at Risk, and I've got their websites here. And there's also a global invasive species database that's available. So there, in summary, it can be a problem, especially to native ecosystems on islands. And best thing is prevention. Don't let them get out of control in the first place. So that's sort of the key message here. So thanks for your attention, and enjoy your gingerbread. If you have any questions, I'd be happy to answer them a little later, or
Now we have
now.
time. And just
Okay.
repeat the question, David.
Okay, the question, I know it's getting near lunchtime and everyone's probably having a growling stomach here, so there's no questions. You can ask me later. I'll be around. Thanks.
Thank you so much, Dave. Another
You're
round of
welcome.
applause. Thanks.
Oh.
It is time. Good afternoon. Good afternoon, everybody. Hello and good afternoon. It is your host, Russell Galante, inviting you back. Once again, for the nth time today, we have a global authority on one of the members of the Order of Zingiberales. We've got Gabriel Sachter Smith. So Gabe is an independent banana researcher as well as a full-time banana farmer on the North Shore of Oahu. He began studying bananas at the ripe age of 14 years old in 2004. He received a bachelor's degree in tropical plant and soil science at University of Hawaii at Manoa, as well as a master's studying banana bunch top virus, one of the most important viruses in bananas, host resistance, and exploring new varieties for farmers in Hawaii. But he's most interested in varieties and breeding in bananas. Gabe is really well known for his knowledge in bananas. I'm very excited to have him here, so thank you.
Thank you.
And then
All right. Okay, aloha everybody. Good morning or good afternoon. Thank you to the organizers for having me here. I'm always excited to talk about bananas, especially in a room full of people that also appreciate it. So what I'm going to do today is give a sort of brief overview of my personal travels and bananas that I've seen along the way that I think are interesting to share with you today. This is not nearly comprehensive of all the bananas out there, nor is it comprehensive of all the bananas I've seen. But what I'm going to try to focus on is to give an overview of the diversity of wild species, and then also touch on edible cultivars and especially how they interact. And it's been something that I've thought about for many years is what am I more interested in, wild species or cultivars? And I've never thought I had to decide, but I realized that my biggest interest is how they relate to each other and you know, the dynamics between them. And the slide is not advancing.
let's see if we can change it. Okay, try now. It just didn't advance. There you go.
Okay, perfect. So yeah, you got a little bit of background, but I started studying bananas when I was around 13 or 14 years old in Colorado. I grew up at about 8,300 feet in the Colorado Rockies, about as far away as you could grow or be from where bananas grow. And maybe that kind of catalyzed my interest in them as this very foreign, exotic thing that I wanted to learn more about. I grew them as house plants. I did science fair projects in high school. I began emailing researchers and just reading everything I could. I volunteered at the Denver Botanic Gardens And I just grew a lot of different plants and read about them all I could until I got the opportunity to come here to study them full time. Now I'm a banana farmer, so there's some fruit there too. And as much as I have to focus on just producing as much as I can of the most straightforward, commercially viable varieties for economic reasons, I still can't help myself from being interested in diversity. So as you see on the right side of this slide, this was actually just a little sample harvest for our annual banana festival that we host each year. So I maintain around 150 or so varieties at any given time. That includes wild species, cultivars, not including hybrids that are in development, because if I start counting each seedling, you know how that gets. But I do try to keep around some diversity for my own amusement, really, mostly. And here's just a little sample of, a very small number of the different types I grow, but you can see already just the difference between them. And this is just edible cultivars. So as we know, the Musaceae, the banana family, is closely related to Heliconias and all of our other friends being in the Zingiberales. And I'm well aware that this artistic dendrogram is extremely outdated and inaccurate, but I just think it's really pretty, and I would like there to be an equally pretty, updated, taxonomically relevant version. But yeah, of course, it's related to all those others. And there's 3 genera recognized with around 100 species known currently. And mostly they're native to Asia with a little bit in Africa. So the first genus that we'll look at is Inseti. This is a relatively small genus, only around 7 or 8 species, kind of varies. And it's native to Asia and Africa with a number of species in both continents. An interesting thing about them that's a little bit different from classic bananas musa is that they only flower once and then completely die. And there actually are domesticated forms for food in Ethiopia, but not for the fruit. They're cultivated as essentially a starchy root crop. And that's Inseti ventricosum. Unfortunately, I've never got to actually see the cultivated forms. They pretty much only exist in Ethiopia, and there's relatively little information about their diversity and how the different cultivars vary between one another. But there are a couple selections that are really, really common ornamental plants that you'll see all over the world, especially just in temperate areas as a summer annual, especially this red leaf form. So it's always fun to see them. One of the Asian species that's most common is Inseti superbum. This is kind of what it looks like. There's definitely a lot of different forms of it, but they're not that well characterized or described. But as you see, if it's in the ground, that one is actually flowering on the Big Island, Hawaii Tropical Botanic Gardens. From the ground to the top of that inflorescence is a good 20 feet. But if you grow it in a pot, that one is at the National Botanic Gardens in Washington, D.C. It's only about 3 feet tall, so kind of adaptable, kind of interesting. Inseti glaucum, this is probably one of the most widespread species, native throughout Asia, all the way from India and even through to Papua New Guinea, you still find them. It's definitely cultivated as well as being wild, but typically when it's cultivated, it's either for ornamental use, sometimes animal feed, or sometimes the seeds are used medicinally. One thing that I learned about them that I'd never seen in any publication or any types of photos before I got to see them up close is that the fruit is pink. The pulp, it kind of has this like pinkish thing to it and actually tastes really, really good. Kind of like Smarties candies. Unfortunately, the seeds are about 1 centimeter wide, extremely hard, and don't make for any actually fun eating experience, but like a professor of mine, Skip Bittenbender, some of you may know, said it's a good fruit for monkeys and children. There's a little bit of pulp that you can scrape around there. They also have these really cool, beautiful green inflorescences that are quite interesting. And like I'd mentioned before, Inseti are known for being truly monocarpic. They typically don't make any suckers, although in ventricosum, the cultivated forms, you can induce suckering artificially. But left to their own devices, they flower once, they produce seed, and then completely die. So that's what kind of they look like after that. So the second genus that we'll look at is Musella. This one is kind of debated. It goes back and forth depending on who you ask, whether it's its own genus, but it's closely allied with Inseti, genetically very close. There's only really one species recognized, and actually it's almost extinct in the wild, and it's thought to be extinct in the wild for a long time, but it's really widely cultivated, especially in Southwest China. It's used as like animal feed, and it borders like rice paddies and stuff. And it's native to kind of highland, dry areas that are very cold. And so you'll find it growing in places where there's no other bananas that can grow. They're very, very short plants. You know, a large specimen with the leaves, you know, might be 2 meters, but the pseudostem may be like 1 meter tall. And they have this beautiful yellow inflorescence that's sometimes called the golden lotus. And in some Buddhist traditions, you'll hear about this plant, the golden lotus, and it's this one, and usually in that sort of area of Southwest China. A few years ago, there was a newly discovered wild population found in Sichuan, China, which if you imagine where bananas would be, that's very, very far north. And it's kind of this red-orange bract form. So that was named as variety Ruber-bracteata. It's very beautiful. And in China, actually, they've been breeding with it to try to produce these ornamental varieties. And what's interesting about them is they're basically succulents in the way that they grow. When they flower, they're deciduous. They drop all their leaves. And you can even dig out the flowering plants, shove them back in a pot, and they'll continue to flower and bloom for sometimes up to a year or more. And so the way I look at it is like, sure, it's an ornamental plant and sort of a cut flower, but it's almost like a living statue. You can grow them in the field, dig them out, put them in a pot, and then put them on your doorstep and just have this continually flowering little banana statue for up to a year or more sometimes. These are just some different selections from, this is a breeding program in Yunnan. And you can see just kind of how interesting and fun they look. And especially in all the hybrid combinations, this is the top-down view of them. So many different things you can do. Unfortunately, as far as I know, this Ruber-bracteata form, the red form, is not available in cultivation anywhere outside of China, or probably someone has it, but I don't know them and I don't have it. But it's just a really cool plant. So the diversity of bananas is never ending. Oh, and another interesting thing about Musellas that's a bit unusual in the genus, or sorry, in the whole family, is that they'll produce these short, leafless, lateral inflorescences, not unlike some gingers. And it's pretty common. So they'll flower with a main terminal inflorescence out the top of the pseudostem. And then where you would normally have more keiki come out, they'll still make keiki, or sorry, suckers, but also they'll make these little mini inflorescences that just jut out everywhere. So it's kind of can bloom all over the bottom of the plants too at the same time. Okay, now we're going to focus on Musa, which is the most famous and widely recognized genus of the family. Musa is native pretty much exclusively to Asia. There are populations that have popped up in a couple places. There's one off the coast of East Africa. It's unknown if it made it there by itself. There's and it extends a little bit into the Pacific too. There's around 80 species recognized, but the taxonomy, like many things, requires a lot more work. And I was very inspired by Dr. Kress's work before, because I've been thinking that same type of work and layout could very easily apply to Musa, but hasn't happened yet. There are currently 2 subgenera, Musa and Calimusa, formerly 5, and they're kind of divided up somewhat geographically, somewhat morphologically. Recent genetic work has shown that there's a lot of species that didn't really fit into either one, and so it was simplified to the 2. So Musa section, this is the most sort of common and typical portion of the genus. It includes almost all edible varieties, the wild species that the edible varieties are domesticated from, and it's native to Asia and the Pacific. It was formerly U. Musa and Rhodoclamis. Musa velutina, that was in a presentation earlier, is a member of this one. There's other these are some former rotoclammy species, Manii, ornata, rubra, or laterita. These are species that are typically smaller. The inflorescences tend to be erect, although it's variable, but the bracts are really typically brightly colored. And the reason for this is that they're adapted to bird pollination, as opposed to some others that tend to be, more duller colored bracts that would be bat pollinated. And I think there's a lot of parallels between the evolutionary history of Heliconias and Musa, and especially when Dr. Kress was talking about those Melanesian species and that are the green bract ones, it occurred to me, I was like, the wild Musa in that same region have also become green bract, which is a little bit unique in the genus. But there's definitely a very, to me, obvious but untapped pool of knowledge there about, how these plants are interacting in their environments and shared pollination biology and things like that. This is just an example of a species that was previously only known in cultivation, Musa rubinia, but on an expedition in Yunnan in I forget when that was, 18 or 19, we did manage to find some wild populations of it. And interestingly too, I was noting in the Heliconia presentations how inflorescence position, whether it's erect or pendant, seemed to be a really strong trait. In bananas, I'd say it's loose. And even within like this rubinia, they might start out erect and then go horizontal and then go pendant and then go back to erect. Or even within the same mat, the size or the age of that exact shoot might dictate whether it's going to have an erect or horizontal or even pendant inflorescence. So it's quite variable. We also found orange forms, which is not unusual. And honestly, seeing it all, I actually thought, well, this is probably the same as Musa manii, maybe Sanguinea, which is sort of a imperfectly known species. But yeah, this was the first real photographs from the wild of this species. So that was pretty exciting for me personally. Okay, we're just going to kind of jump around to a few different wild species and then tie them all back together. This is Musa itinerans. It's a really, really widespread species, especially in northern Indochina. It kind of exists, well, India, Myanmar, southern China, through to northern Thailand, northern Laos, northern Vietnam. This is the dominant, most common wild banana that you'll see. And it's extraordinarily diverse. There's been something like 20 plus botanical varieties described of it, but just being in the field with them, I'm pretty sure that they're just, even within a population, they're so diverse in their morphology that I almost think it's worthless trying to describe them as these botanical identities, because they just they're all over the place. This is just even from one small roadside vendor actually selling them for medicinal use, the seeds. Let's see, these are from the same area, and you can see this, even the variation in fruit color. They're called itinerans because they have this itinerant or running suckering habit. And when you see them in the wild, they won't form a clump like a typical banana. They'll be sort of these widespread, sprawling groves. Sometimes the rhizomes will travel up to 5 meters underground before they come up. And some members of this species are extraordinarily tall, like 40, 45 feet plus. But that's kind of what they would typically look like in the forest. And another interesting thing about them is they're not cultivated hardly at all because they're just so common, but they're extremely widely used. So the sap of them is not astringent or bitter at all, unlike a lot of wild, or pretty much all other bananas. If you were to just take a bite out of the plant, it kind of has that green, unripe banana flavor and sensation to it. This one, basically nothing. And so the flower buds are very commonly used as a vegetable raw in salads. The fruit is extraordinarily commonly infused into alcohol in Vietnam. I had read about this, but I did not understand until I went there just how common this is. Literally every single restaurant will have it ready to go at the front of the restaurant, and almost every single household too will have their own batch. And in every single village, you'll have a medicine shop with piles of wild bananas, often itinerans, processing the seeds, extracting them, and selling them. So it's a really, really strong part of the local culture there in northern Vietnam. And the inner stem, this is like the flowering stem, is often used as a vegetable too. So it's a really important plant culturally, but it's just a wild banana. This was one of my favorite banana-based dishes I've ever had in Yunnan. This is actually young itinerans leaves that are still rolled up in the pseudostem before they, are exposed to the sunlight, and they're white and tender, and they're chopped up, and it was kind of like a noodle salad. So again, this is like a non-astringent species and really tasty. Okay, Sycamensis. This is a species that was only previously known and still is only officially known from Sikkim, or like the foothills of the Himalaya region in India. But a few years ago in China, some different wild bananas were discovered and kind of classified, but the classifications to me didn't make a whole lot of sense. And when I was there in the field, I actually realized, whoa, these are, I think, Sycamensis that are in Yunnan. And they're just kind of interesting species. They have a few different traits that really are a little bit unique. They almost always, lose their male bud and then lose all their leaves, and the fruit just hangs there for, I don't know how many months. I don't know what they're waiting for, but they're native to these typically highland kind of cold areas, even in areas with annual frosts and snowfall. So they could potentially be used in breeding for cold hardiness. But they just have this very interesting look about them. And I was also noticing that in China, there's especially some that were previously described as the species Dichongensis, which was then reduced to just being a form of Balbisiana. But I realized that the papers that did that, like, I won't bore you with the details, but completely skipped over some really important taxonomic or like morphological information, and basically just wrote this species out of existence. And I, looking at it again, I was like, oh, I think it's still there. It was still good. So again, just more work to be done. But yeah, just kind of really fun looking stuff, really. And like I said, they're native to really cold areas typically. So this is, I forget how high elevation this was in Yunnan, but they're growing in areas like conifer forests with wild rubus and things like that, in areas with snowfall. And so it's just a really sort of unusual thing to be in these areas and then see wild bananas at the same time. This is one of the species too that's really commonly or more commonly cultivated in temperate and cold areas of North America and Europe because of its cold hardiness. And kind of like with the itinerans, it's not cultivated, but it is often harvested as a vegetable. In this case, the flower buds are commonly used too. Okay, so the species we're going to talk about, Balbisiana, this is one of the main primary ancestors to most edible bananas. I'm starting with it first, but it's the secondary ancestral parent. There's a lot of diversity of it. But what's interesting about this is it's essentially a wild plant, but it's very difficult to find in the wild. You almost only can find it in cultivation, either grown, like I was mentioning, for seeds medicinally, or for animal feed, using the leaves as wrappers. So it's a very, very, very common plant with a lot of diversity, but wild populations are extraordinarily hard to find. And even there's two types of cultivated ones. There's essentially the wild type cultivated, which has small fruit, normal seeds. It looks and appears just like any wild banana, but it's still cultivated in gardens. But then throughout Southeast Asia, you also have this form that is domesticated. The fruit is a lot larger, the bunches are a lot larger, and it still requires pollination and makes seeds, but the seeds stop developing partway through. They typically don't have an endosperm or embryo, and they stay soft basically. And so you can eat them like an edible banana, even though they're seedy. And it's this kind of interesting case of like, well, it's sort of a whole different beginning lineage of banana crop domestication, but it's sort of at the early stage, which is interesting to see. And they still will use those seeds and fruits in the medicinal alcohol. But you can, see them compared side by side here. And this is an example too of one of those medicine shops I was describing where in almost every village in northern Vietnam, you'll see this, just piles and piles of seeded bananas that are highly valued and being ready to be processed. Okay, so the primary ancestral species to edible bananas is Musa cuminata. Historically, it was divided into it just was many different species that have over time been kind of lumped into all kinds of subspecies and varieties. And it's really complicated as a sort of taxonomic entity. And I think personally, this is not published, there is a lot of justification for resplitting it back up. But because it is the primary ancestral sort of population from which edible bananas came from, it would just screw up so much of people's understanding of bananas. It would just have a lot of ripple effects. So for now, it's kept as it is, even though it has an extremely wide distribution. It's extraordinarily diverse. And we can look into a little bit of the genetics here that you can see just like how ingrained it is. There's lots of different forms throughout Southeast Asia and in the Pacific too. Subspecies Banksii, though, is probably one of the most important. It's mostly native to Papua New Guinea, although the populations, depending on, they're not all well studied, and bananas are often in very difficult to access places to get well, usually they're just growing on the side of the road, but the countries, the islands throughout Southeast Asia are difficult to access. But they might occur in a little bit of Eastern Indonesia. They typically have very large bunches. They're really big, robust plants with large fruit. There actually, though, is one population in Samoa, Fa'i Taimane, which means like bird poop banana, because the birds will eat the fruit and spread it around. It's a little bit, it's still unknown whether it got there by seed or as a canoe variety that Polynesians brought there, but it doesn't exist anywhere between New Guinea and Samoa. So that's a question. This was sort of like a holy grail moment for me personally when we were in Samoa doing these expeditions. We came across a specimen of this wild Banksii up here that was parthenocarpic. So parthenocarpy, meaning virgin fruit, where the fruit just forms pulp, not instead of pollination, but regardless of pollination. So the fruit could still be pollinated and set seed, but having that trait where it'll form pulp anyways is really the first thing that needs to happen for domestication. We found a essentially wild specimen of Banksii that was parthenocarpic with just a very couple seeds in the fruit. This was sort of a standard one. And even though this looks like it has a lot of pulp, it's just because the knife sliced, in that thin layer of pulp. But this fruit would be about 95% seed, maybe we say 5% pulp. This one is probably like 98% pulp. But just seeing that plant there, it was so easy to imagine that, somebody 5,000-10,000 years ago was like, hey, this one's a little better. I think I'm going to take a sucker and bring it back to the village, and you can actually, start the cultivation. So it's a little bit like time travel for me, and seeing what that looked like in that very early opportunity to make a selection towards domestication. Okay, so continuing on with the cuminata though, Malacensis, this is a really important species or subspecies also for the domestication history of edible varieties, and it's native to Peninsular Malaysia. It typically is horizontal bunches with a red bud. But recently I was in Peninsular Malaysia doing some field studies and kind of really got to see what the diversity was like. And like anyone who's worked with taxonomy and wild plants can relate to, is that it's never simple, but it's worse than you think. There's been a long-standing variety called, I don't know what, Musa cuminata variety flava, which has been known to have a yellow bud. To me, it's pretty clearly just yellow budded forms of Malacensis, which is a bit controversial. But if you go in the forest and you look at these populations, you'll have plants that are nearly identical. One with a red bud, one with a yellow bud. These are separate photos, but these are literally growing about 15 feet apart from each other in one population. So we can see in at least Musa, it's not controversial to me at all. That bract color really doesn't mean much. It's a nice guideline, but there are lots of species known already that variations in bract color are just written into the species description as, yeah, it can be red or yellow or brown and green. And then some where that bract color, kind of like the Rubinia earlier, is kind of the only justification for it being a different species. So again, lots of work. I'm not proposing answers here, just observations and all the problems that exist. There is another population of acuminata on Peninsula Malaysia, subspecies truncata, which is typically only found in the higher elevation areas. And again, it's not perfectly known either, and there is a pretty clear gradient between it and the Malacensis forms. And as you literally go up the mountain, you'll see it slowly change with a bunch of intermediate forms. Bananas typically, you know, can pollinate interchangeably fairly easily. So it's not always easy to distinguish exactly what is a species. And I was also thinking too, why there were things described in the past that to me now are so obvious as being incorrect. And I realized that a lot of these taxa were described at a time when it took so long to travel. You know, you might stop in one place. Like I'm imagining, you know, these taxonomists in like the 1930s and 40s, they stop at a village, they explore the plants around there. They're probably exhausted. They collect a few samples and all right, we're going by river or by boat up the coast to this other place. And then they see something else and it's totally different. And like, okay, new species. Well, now we travel in cars and we can stop as frequently as we want. And there's rest stations with snacks. And so we can look at everything. And it's like, actually, this is all just one big variable population, and putting those lines between them maybe made a lot more sense in the past, but it's a bit different now. For example, too, this is one that when I first saw photos of this, I was really excited, like, oh, maybe this is something really, really new in Peninsula Malaysia. And it even has a sort of tentative name, acuminata variety Berdiri. And again, they come in red or yellow forms. And at first you think, wow, this is really distinct. It's their plants are shorter. The bunches are erect as opposed to the typical Malacensis, which would be more horizontal or pendant. But again, you can also see every iteration of bunch shape and fruit size and bract color between this and the typical Malacensis. So where you draw the line, unknown. This is just another subspecies of acuminata worth noting. Bermanica, it's one of the primary ones. I think there's, like I said, maybe 20 or so different taxa of acuminata described, but the ones I'm highlighting here are the ones that have been shown to contribute to the domestication of edible bananas most prominently. There's classic types, which kind of look like this, and then one that was described as acuminata variety chinensis, which is a waxy form. But again, in the field, you can find waxy and non-waxy forms growing within the same populations. Okay, we're going to start a transition into edible varieties for a bit. This is a graphic from Simmons, The Evolution of the Bananas, which is sort of a classic banana text. And what's amazing about this isn't that it's accurate, because this was produced in like the maybe 50s or 60s. There's a lot we know now. But what I'm always amazed at is actually how close to accurate it was, using no real molecular tools or DNA. It was just based off pure observation. Essentially that you have wild acuminata, wild Balbisiana, selection for Parthenocarpi from the wild acuminata that are then crossing with the Balbisiana and producing all these different groups. That's more or less accurate, which to me is just mind-blowing that someone could figure this out just by sort of looking at plants, thinking about it, and then 70 years later having the genetics more or less back it up. So that's cool. They must have been onto something. So in edible bananas, we have what are called genome groups. They're based upon ploidy, which is the number of sets of chromosomes, and then the relative contribution of the wild species to them. So you have these groups AA, AAA, quadruple A. These would mean it would purely acuminata-derived diploids, triploids, tetraploids, combinations with Balbisiana. And it's a way to classify them, but it doesn't mean that things within each of those groups are necessarily closely related to one another. But diploid cultivars tend to be really slender. They often have more upright leaves, and they're usually smaller, but sometimes not. Triploids tend to be a little bit more vigorous, a little bit more robust, often with more horizontal leaves. And then tetraploids tend to be extremely robust plants. What's interesting about the tetraploids is that they've been known in banana breeding programs for a long time, but it wasn't until very recently that we discovered some plants in Papua New Guinea that it was shown there are naturally occurring tetraploids, at least naturally occurring in the domestication sense. They don't come from breeding programs. They're just traditional landrace varieties that are tetraploid. And this is an example of one called Buka. And if I was standing here, I would only be about like this tall. These leaves are massive. They're probably about like 5 or 6 feet wide and maybe 15 feet long and almost touch the ground the way they bend down. So just kind of a cool plant anyway. Okay, so within edible bananas, we also have the subgroup concept, which is essentially that you would have a single original seedling, and then that seedling mutates somatically into different cultivars. And then all those different cultivars are collectively known as a subgroup. And there's a lot of different ones. Not all cultivars are assigned to a subgroup yet. And, not all subgroups, really, we don't know all the members of them. There's always more to be discovered. But generally speaking, cultivars within a subgroup share really important traits like disease resistance or fruit, eating quality, things like that. But they vary potentially in size, so dwarf versus tall, pigmentation, whether they're more red or green or combination thereof, and then sometimes in fruit shape. So I'm just going to go through a few of the common subgroups just to kind of get a sense of it. Sucrier, this is one of the most common throughout the world, and it's a diploid. If you ever had or seen like the baby or mini bananas in the grocery store, it's this, and it's a diploid. So I guess that's one little thing. You often hear that all bananas are triploid and the commercial ones are triploid, which is sort of true, except for this, which is one of the most common varieties in the world. It's not triploid. There is actually a Hawaiian variety called Hapai, which is a member of the Sucrier group. And it's the only one that made it this far in the Pacific. And it's called Hapai, which means pregnant in Hawaiian, because about 25% of the time, the bunch will stop midway through the pseudostem, start developing, and you have to give it a C-section in order for it to develop correctly. It'll has to flop out of the pseudostem. And in any other case, that would be considered a, deleterious mutant that you want to get rid of for production. But because it's a culturally relevant thing, it's kind of cool. So that's Hapai actually when it looks normal. Mshare, this is a fascinating group of diploids from East Africa. There's a few cool things about them. One is that they taste really, really good. They're one of my favorite fresh eating bananas. And they're only known from East Africa so far. They have not been found in Asia. They have to be somewhere, but with 10,000 plus islands in Indonesia, we don't know which one it could be. They could be on. But their claim to fame is that some member of this subgroup is the ancestral parent to Cavendish, Gros Michel. So Cavendish is the curved yellow standard banana you think of, call it banana no frills. Gros Michel is its predecessor commercially, and our local bananas here that are favorite apple bananas, that group, which is not popular just in Hawaii, but globally popular too, they all are direct descendants from crosses between a Mshare and something else. So there's been a lot of work to try to figure out how does this group of bananas work, where is it actually from, and how to breed with them. And instead of now breeding with, say, Cavendish to create a better Cavendish, the newer strategies involve trying to recreate Cavendish or recreate Cavendish-like things or Gros Michel-like cultivars using share and some other of the related ones. You can see here this is a fairly large bunch, nice big fruit, and still a diploid too. So again, goes against the grain of that diploids are smaller, weaker plants always. So Papua New Guinea, I've had the great pleasure to study there. It's so diverse. It's insane. Every time I've been, we discover new things. I like to think about it in sort of like the orders of magnitude of diversity difference. So I know the bananas in Hawaii quite well. Even in the old days, maybe let's say there was a few dozen varieties in Hawaii, if we round up like the traditional Polynesian canoe varieties, a lot of them are extinct. And let's say if there's 20 left, that's some level of diversity. Even if you go today in Hawaii and go through every single botanic garden, every single private collection and find as many varieties as you can, which I've done, I might be able to come up with like, 10 that are common, 20 that are a little bit more rare that I could find. And then in total, maybe like 50 or 60. If you go to any single village in Papua New Guinea, you're liable to find between maybe 50 and 80 varieties. And then you go a few miles down the road and you might find 50 more different ones. And part of the reason for that is because that wild Banksii is from there, and that's one of the most prominent wild ancestors, they're still crossing with each other. They're still crossing with the wild stuff. And it's a very fluid situation genetically. And also they've just been at it a long time. Some of these, could be some of the first instances of human agrarian domestication. There's evidence of bananas being cultivated different from the wild ones up to 10,000 years ago in Papua New Guinea. So they've had a long time and a lot of diversity. And there's almost nothing you can say about the bananas of New Guinea other than they're just all over the place. And even so far as that, I think there is definitely, of course, a role for banana breeding. But if you wanted to just create some really interesting, weird banana, it'd probably be easier just to go to New Guinea and find it because it's probably there already. These are just more examples. And what's fascinating too is these are all exclusively derived from Banksii. So in New Guinea, there is actually contributions of some of the other Southeast Asian species in there, which we know then is proof that bananas were domesticated in New Guinea, traveled throughout Southeast Asia, crossed with other things there, and then also came back to New Guinea because there's genetic signatures of wild populations from all over Asia. But most of these ones are purely Banksii derived. So you can see almost the entire diversity of the genus being expressed through just this relatively small wild genetic pool. These are a few of the favorite ones that I found. I've actually had a long-term project to try to breed really short little mini home garden bananas. And again, or you can go to New Guinea. This is called a puka biang. It's about 3 feet tall. From planting to harvest is about 4 or 5 months. It's perfect. It's cute. Yeah, it's edible. Yeah, not big fruit and not a big bunch, but it's cool. And then you also have, this is called pagal. This is another diploid, which the size of the fruit and the size of the bunch rivals or exceeds a lot of the classic plantains, which are triploids and some of the most important bananas in the world. Its culinary traits might be a little different, but again, from a breeding perspective, if you say, oh, we want really long fruited diploids for breeding, go to New Guinea, they're there. This is actually a video which won't play here because this is the file type is wrong. But if, most of you might be familiar with the arrangement of banana fruit in a bunch, which, is pretty much the same as Heliconias, where you have distinct hands, which are the clusters of flowers that are at each bract. There are a few mutants in the edible side of bananas. This one called uriwasi, which means spinning. And the entire, it's one hand of fruit that actually spirals around the whole bunch. And then even when it transitioned to the male flowers, it just keeps spiraling down. And I was thinking before I saw this plant, because I knew that it existed, it's like, how does the bracts in that work? Like if you have one continuous spiral of flowers, and the way it works is as weird as you think, there's one bract. And it is also spiraled. and it just sort of falls off and sloughs off as it dries and just is this chaotic, weird thing of a never-ending bract with a never-ending line of flowers. So kind of cool. Probably if you studied what mutation that is, you could learn something about the Zingiberale floral morphology evolution or something. Okay. I think the motto for the tourism agency in New Guinea is Papua New Guinea, land of the unexpected. Definitely is true. It's been a while, maybe 15 or almost 20 years ago, there was this plant that came on the horticultural scene in the banana ornamental world called Siam ruby, and it's a sort of like red leaf thing. Nobody knew really where it came from, or at least the information wasn't available. Like any rare new plant, it was really expensive, like $2,000 a plant. Then it came down once it got into tissue culture. And when I was an undergrad, it was sent to a conference in Ohio, the Ohio Short Course or whatever. That year, I was talking to some folks. I saw it everywhere. They had it as a bedding annual, that it was like the most popular, like #1 selling new bedding annual plant in the country that year. It was this red leaf banana. In 2016, when I went on my first expedition to New Guinea and Bougainville, it turns out they were there. And people were just growing them as these ornamental plants. But we didn't find just that. The Siam ruby kind of looks like this one, but actually 5 different color leaf forms. What's fascinating about these is they are definitely domesticated in some way. They almost never flower. I've only seen one example of any of them flower. They make a few flowers that don't even seem to have like pollen or be able to set seed. But they're grown in home gardens. And we ask people about them, like, where do these come from? They're like, I don't know, we just found this one in the forest, or I just got this one from wherever. But they really, it's come out that in their genetic analysis, they don't really relate to anything. It's like they're their own group. We don't really know where they came from, how they work, what's going on, but they're really, really pretty. And there's one in particular, which initially I just called the rainbow variegated. And there's a little bit of a, sad story with it for me personally. It was the end of a collecting day in Bougainville. It was just about sunset, and we were about to leave this village, and everyone was really tired. And I was like, what is that? I saw literally this exact plant across the garden. I was like, I've never seen a banana like that. This is going to be like a major thing. And everyone was like, wow, that is cool. The people that were growing it were like, oh yeah, that's just some thing. I don't know, whatever. The fruit's not good, so we don't care about it. And I was like, can we collect it? They're like, yeah, please get it out of here. So we collected it. And when we asked the names, most of the banana names, like cultivar names, are just made up on the spot. It'd be like if somebody shows up at your yard here in Hawaii, or I don't know if everyone here is from the tropics, and you have this coconut in your yard, and a coconut scientist is like, what's the name of this coconut? And you're like, I don't know, it's a coconut. But yeah, but it's a little bit different. It's kind of yellow, and the fruit shape is pointy, and you're like, I don't know, pointy yellow coconut. You're like, great. And then that's written down, and it's become this official thing. So this plant didn't have a name, but somebody said, no. Well, why is it called no? Well, one of the words for breast, like a woman's breast in the local language here, was no, because they said the fruit kind of looks like that. And then someone else said, no, it doesn't even make fruit. And then someone else said, no, the fruit, it makes fruit, but it's not good. And then we're like, whatever, we're just interested in collecting it. But the name no stuck. And that's what we wrote in our in our field notes. And when the publications came out, because normally we're collecting with, officials from their national collections to add to their national collection, and then it's backed up in international collections in vitro typically. So, we have publications, and I knew that was like, whenever these publications come out, people are going to come get these plants. And so what we were trying to do is to work with, and this is why it didn't work, some international NGOs, the local communities there, the New Guinea, national germplasm situation, to try to, can we monetize these on the market and have the money go towards New Guinea conservation, banana conservation, or go towards these communities that they came from before they're just come and plucked out? And the answer was we tried, but we couldn't. And starting about 2 or 3 years ago, maybe 2 years ago, these start popping up on eBay. They have the name No-No, all these other varieties too that we found. And yeah, people just followed our publications and bought tickets and got the thing. So it's a little bittersweet for me because now I've seen it around. And I'm like, man, I was literally, I don't want to say I discovered it because the people who've been growing this for how many hundreds or thousands of years discovered it. But I was like, I actually did kind of like see it and say, we got to get that banana. So yeah, it's interesting now, but yeah, really pretty plant really is the moral of that story. Okay, we're gonna go back to some cultivars for a little bit because that was like in New Guinea. So Cavendish, this is the quintessential banana, and it really is like misunderstood. I'd say the coolest thing about Cavendish, there's a lot of diversity to it, but it's actually a traditional landrace variety, and I think that's a little bit unusual among edible crops. Is that this is like the #1 most consumed fresh produce item in the world and #1 most traded. And it is a landrace variety from like northern Vietnam, basically. And I don't think many other crops have that. And so a lot of people look at Cavendish as being the sort of junk banana that you want something else. But I always look at it as like, wow, this is an amazing banana that it made it to that level. And this is it just growing in someone's garden and pretty much the same and at a local farmer's market in southern China. Okay, Gros Michel, this is the previous one, the commercial predecessor to Cavendish. We're just gonna skip through actually this because we'll get to, I wanna get to this part that, this is some bananas in East Africa where they are extraordinarily important. In some dialects of Ugandan languages, the word for food and banana is the same because if you didn't eat bananas, you didn't eat that day. So just some photos from around the world. They also have these traditional varieties that they'll make a fermented beer-like thing from, or they even have cultivars that are specifically domesticated for that. They're a little bitter, a little astringent. They don't taste that good if you eat them by themselves, but when you ferment them, they make a nice party drink. Plantains are a specific genetic group of banana. They're not a different type of really crop at all. But there's 3 different types: French, false horn, and horn. Ken has been asking me for these horns for a little while now, and I'll get them to you soon. But they're kind of interesting in that they typically just make 1 or 2 hands of very, very large fruit, sometimes 2 or 3 feet long. Mauli Populu, this is a Pacific Island group and a representative of some of the Hawaiian varieties. Okay, I'm going to skip through these cultivars because I want to get through actually a really kind of back to what I was hoping to get. This is just male bud diversity. It's really cool. We always think about the fruit and bananas, but I guess this crowd might be thinking about the flowers too. And I always pay attention to the flowers. And these are all from edible cultivars. So it's another, point of distinction. How we identify them in the field is not just by the fruit, but also other parts. Okay, so back to wild bananas for a minute here. Cali Musa. So that was all section Musa that I went through. Now we're going to section Cali Musa. There's a bunch of different wild species, native mostly to Southeast Asia, a little bit into the Pacific. They tend to be really colorful, upright. These again would be kind of bird pollinated ones. But then in New Guinea, you have ones that tend to be more bat pollinated and start to become green, kind of like those Heliconias. There's Indo-Chinese Cali Musa, which are all closely related, some of them with persistent bracts, not unlike Heliconias. The taxonomy between these is a little mysterious, but they definitely form a group. They're harvested in the wild commonly and used as cut flowers. This is in Sapa in Vietnam. Okay, this is a New Guinea species, Musa maclay. They typically have erect bunches, very high carotenoid levels in the fruit, making them orange, and purple sap. Really cool. And it's the probable wild ancestor to Fetty bananas, which we'll get to in a minute. This is Buchensis, which is very closely related to Macleayi. And as you can see, kind of variable between purple or green buds. This is a potential Buchensis that I found in the Solomon Islands, not yet described. Green buds here, huge bunches, but really small fruit. definitely in the Buchensis maculae variety erecta sort of complex there. Okay, but these are sort of the ancestors to what we are called Fetty bananas. So we talked all about the edible varieties that came from Acuminata and Balbisiana. There's a completely separate but parallel domestication lineage called Fetty. This is a wild maculae. This is a domesticated Fetty. Orange flesh, really high carotenoids, upright bunches, a lot of cultivars, but very poorly understood. This is an example from Samoa, some more stuff from Samoa. And Rarotonga, they still exist. They're cultivated, but they're almost regarded like wild plants in that there's these huge patches planted up in the mountains, and they could be hundreds of years old, and people generally just go harvest from them, but they're not actively cultivated very much. But they're definitely very domesticated. There's some diversity from Rarotonga that still exists. And a lot of these we thought were maybe the same, but then when we actually did the genetic work, it turns out there's a lot of diversity within the two. So they could still be crossing and having some seedlings generated, but still a very domesticated plant. Kind of like the wild maculae, they tend to have purple sap, which is quite interesting and shocking when you cut it open the first time. And the pseudostem fibers are usually black, and so they're used a lot to add color to traditional weaving crafts throughout the Pacific, and also the sap used to dye tapa cloth. The flesh, again, super high in carotenoids, really rich, very tasty, and completely different texture and taste than pretty much all other bananas that you would have had. Now, Schizocarpa, this is a species from New Guinea, which is interesting in that for a long time, it grows with Banksii, but not known to cross very much, although there are some wild hybrids. But there's actually cultivars that have been known for a while that are direct hybrids. So this one we call an AS, this is an AAS. But what's interesting with these sort of next generation genomic tools, we found that almost everything actually has Schizocarpa in it, which means that it probably crossed into Banksii at some point, and then those ones generated all the other edible varieties. And so even in something like Cavendish, there's Schizocarpa in it as well. And these are plantains also with Schizocarpa. So the sort of trope of bananas come from these two wild species, Acuminata and Balbisiana. It's definitely at least 3, maybe more, because some stuff we're even finding has big portions of genomes that we don't know what they are. The wild plants haven't been found yet. Itinerans is an example, like we talked about before. It grows among all these different edible varieties in Indochina. But a few years ago on an expedition, we found some plants that I didn't recognize them. I didn't know what they were, but they were unique. Turns out they're itinerans hybrids. So this is a tri-specific, completely new genomic group of bananas that we found. And the genomic work of that showed that, yeah, there's this brown bars represent itinerans DNA that are completely intact, full itinerans genome. And if these are growing next to Acuminata in the wild, they never cross. But somehow an Acuminata and Balbisiana derived hybrid was able to cross with them and generate some diversity. There's also intersectional hybrids between Musa and Kali Musa, which don't make any sense according to the textbooks, but yet they exist. Some that are kind of Fetty-like, but with other traits, so they're really high carotenoids. And even some of these tetraploids that we found in New Guinea, they look very similar to common triploids, but they're tetraploids. And it turns out they have full intact Kali Musa genomes in them as well. So these could have up to 4 or 5 different species from 2 different sections coming together to make these edible cultivars. This is all just to say it's very inspiring. I do banana breeding as a sort of side hobby, which that could be a whole nother presentation, but all of this has really just inspired me to expand beyond the classic banana breeding pool in order to push the limits of what's possible. Not just for edible varieties. So these are some of the edible stuff I've been generating and messing with, but also ornamental stuff. These are some on the way to edible varieties, but they looked kind of cool. So I selected them for ornamental. And then even within Kali Musa, I've started doing some hybrids. So these are some interspecific potential cut flower varieties that I've been working on. Anyway, I know that was a lot and I blasted through it, but thank you so much for your attention. And if you have any questions, I guess I'm here now. I don't know if you have time. And then feel free also to email me or check us out on Instagram if you have any questions. Thank you. Yeah. There's definitely, oh yeah, so the question is like, yeah, in the village, are they eating all of them? And the answer is mostly yes, but they're not necessarily always have specific uses, but some of them do. But what I find most interesting is that a lot of the reason they might keep a variety and the uses for them, they could be edible, but they're not related to its eating quality. They have really deep cultural significance. So some varieties might be, like there's one in Bougainville that in this tribe, a plant of that is given to young girls during this womanhood ceremony, and then they keep it their whole life, and as long as they have it, they're good, and then they pass it to their daughters. Or there is another one that's planted on the edge of their village, or like the edge of their, I guess, territory, because this other tribe believes that there's evil spirits in that banana. That tribe didn't, but they say if we plant it on the edge and they don't come into our land. So there's a lot of reasons like that too, where they keep these varieties for those types of reasons as well. So yeah, any other questions? Yeah.
Variegated types, does the fruit come out variegated as well?
Often, yes. David?
Tell us about that Kandari food process.
Oh yeah, that's a giant triploid from New Guinea. Yeah, it's cool. It's this massive, massive plant. Sometimes the pseudostems get about 2 feet or more in diameter. In New Guinea, one of the names for it is Papa Ground, which in the pidgin means like father of the ground. Landlord is the other translation of it, because it's always there watching you and it never goes away. It's like it owns the place because they're these massive, massive plants. All right, thank you.
Next up, we've got Maria João. So Maria is an agricultural engineer with a master's in hort science and technology from the International Center for Advanced Mediterranean Agronomic Studies. And she works at the Regional Directorate of Agriculture of the Regional Secretariat for Agriculture, Fisheries and Environment of Madeira, Portugal, where she provides technical support services to flower growers since 1994. So I'm really interested in your work. Her main research is topics are in Heliconiaceae and others in Gibberellales. This is one of my favorite talks that I'm looking forward to seeing. So I'm really excited because it's the type of work that I was good enough to do. So I'm excited to see how you do it, so I can learn a little bit from you. Maybe you can just do a little conversation while I get your presentation loaded up.
Okay, it's okay. Yeah. I'm working as Russell said. Okay, Madeira Island, I don't know, we call it Madeira, you pronounce it in a different way. Madeira Island belongs to Portugal, like Azores Island, and we are located a little south from the mainland, and we are facing in the east side, Morocco. So sometimes our climate is a little changing because we have the winds from the east coming with some sand. Okay, so it's all right. Good. Okay.
Yep. And if you want to stand here, you no problem.
Yes, it's okay. Maybe I can take it here. Huh?
Just talk real close
Yes,
to it.
yes, okay. No, I think I can do it like that. So, because in my service we have a floriculture center where I'm. So in that I'm going to show you, I started my work there on that floriculture center. So I was doing a lot of research more for the growers of the island to have more biodiversity in the in the production they have it there. So one of the ideas, because proteas and it's another plant, but Heliconias are very expensive flower. So it's one that we could supply to the mainland with not so high cost, and it will give some income to the growers of the island, okay? So on 2000, we bought some genotypes from Costa Rica, and I got these plants from my boss. I took care about them. Some of them died. So what it maintained alive, and I put it in the field. I planted in the field. It was more or less 20 genotypes. of Heliconia and two of Etlinger that I'm going to show my work about it, okay? So let's start. So the title of my work is The Yield and Flowering Patterns of some Heliconia and Etlinger genotypes in Madeira Island. Let me see if it's work. So in the last years, why I'm doing I did this work. I did this work for 8 years to see, because the market showed an increased interest for Heliconia and torch ginger cut flowers. I forgot to tell you, my technical support is mainly for growers that are producing cuts, cut flowers or cut foliage. So we needed to study the adaptation of these plants to the Madeira climate, okay? So for that, I saw that there was a need to do it, to diversify their production in Madeira, okay? So I explained already, Madeira Islands, like you see, it's the mountains are going like this, compared with Hawaii. You have Hawaii is like this, or like this, sorry. So our mountain that is going until 1,800 meters up, it's horizontally like this. So it's creating two kinds of climates. So in the north, it's more temperated with more rain. Always a lot of humidity anyway, but not so high as here. And in the south, it's more arid, okay? So what we did is when the Portuguese arrived to this island, it was full of wood. So that's why it's called Madeira Island, because Madeira means wood, okay? So they burned all these trees that were there just to, to settle down there and to make agriculture. And because you could see that orography is so bad, because you have always going up and down, we built terraces to make the agriculture, okay? And the water, because this was a little more arid, but better climate, to live on. We built kilometers of channels that we call it levadas, to bring the water from the north to the south for the populations and for the agriculture, okay? So where is my center located? It's here. Funchal is here, okay? So it's here where it's Ponta do Sol. It's the warmest place of the island because we have a lot of microclimates. And so I'm going to show you, so the South Coast, as I told you, it's subtropical climate. So this is, as you can see, my center. So I'm very near by the sea. We are getting sea salts a lot. So it is destroying a lot the plants, because we have a lot of different species here for cut flowers that we are making some essays and also producing flowers for different events from the government, because every weekend we have some kind of event in the island. So I was doing a lot of experience, not only with Heliconia. So the Heliconia, this is a Strelitzia field, I can still see it. These are the Heliconias, and this down also in this second terrace. This is the first terrace, that one is the second terraces. And the other ones, it was here and on that shade that you can see here, the shading place. These are the places where I made that experiment, okay? So this study was done from 2012 to 2020, okay, so 8 years. So these are the locations. It was in a shelter, some of the genotypes, and these were the conditions in the open field. So I tried to torch ginger genotypes. I named this one as Rosa, but I invented, I must say, I'm very sincere with you. So I call it Rosa, this pink one, okay, this one. And the other one, it was the one that we brought also from, we bought from Costa Rica, and it was labeled tulip black, this one, okay. And the Heliconias, it was 20 genotypes, so I'm not going to say one by one. So if anybody wants this PowerPoint in PDF, I can give you to you later. So you'll give me your email. I send it to you, okay? Because it's too long, it's 20, okay? It's a mixture. So these were the genotypes that survived, okay? So what we needed, what I wanted to get, it was the information, I got the information for each genotype. about inflorescence type, if it was erect or pendant. I put also the different locations, okay, open field and shelter. It was the plantation date, they were different because I didn't manage to get ready all the genotypes on time. So I did three years of plantations, okay, but all of them will be in the next slides. So I also change a little bit the spacing according also to the place, the area that I had available, because not all the time I had the area that I wanted for the plantation. And so that's why I have one table that I'm saying how many plants I had it of each genotype, okay? So what I did, the observations that I did, that I recorded for each genotype, it was the growth habit of them, the height and the area of the clump expansion, and the inflorescence production, because this is the most important factor for the grower, and the flowering period, also very important for them. And because I have the meteorological station there inside the center, so I gather all the data from temperatures, the maximum, minimum, everything I have it in one table, the relative humidity, the precipitation, and the wind speed. Why I'm putting here the wind speed? Because we are in Atlantic and we are getting different kinds of, we are affected by different winds, okay? And some of them, especially those that are coming from the west side, are destroying a lot the Heliconia, and also we have banana also, because all the leaves are becoming splitted. Sometimes you have to anchor the bananas and other plants and even heliconias because they can fall down. So this is important because it's affecting the production. So I recorded all of it. So because it is a lot of work and 20 genotypes of Heliconia and 2 of Ettlinger, I'm just going just to say some of them to not bore you, because some of you are already sleeping. I notice, you know. So about torch ginger, that so I put it where I planted for you to have one idea. They were planted, both of them, in open field. Okay. So I'm putting more or less that you can check. And what I noticed that Rosa, okay, was a little higher than the tulip black, and the faster clump growth than tulip black. And that stuff is important to know how fast they are growing, because our land is so expensive and the growers don't have so much space, you know. Because the areas are the average of areas for production of flowers is around 1,000 meters, 800 meters, square meters, sorry. So it's small and it's difficult to buy because constructions is getting everything, you know, all the areas for. So people sometimes are selling their own lands because they're getting more money and not having, you know, labor to work the land. So that's why we need really to use all space that we have as much as we can. So about the growing of Heliconia, that I grouped in those that have the open growth type and those that have the spreading or open growth habit, okay? So from these ones, the clump height were between 1.5 and 2.5 meters. And because those plants were planted inside that shelter that you saw, and because I had a big problem, because the roof was becoming darker with the years and I didn't change it. So I started with more or less 60% of shade, and I ended with 90% or 95% of shade. So this was too much. So the normal psittacorn that shouldn't grow so high, they were growing very high, and they were flowering less, and even the flowers, the colors, sometimes the Nicariensis was similar to golden torch. Shouldn't be. Golden torch is the yellow flower, but because of the shade, it was becoming like you can see in the photo. So the clamp area that you see there, it's not so high as it's not so, I have to put my glasses, sorry. The clamp area, it was a little it was not bigger than that, even though they have the gross habit of spreading, because we needed to take those that are coming in the past walking ways. Because if not, we couldn't work on it, because we are harvesting every time, because every week we have some kind of fests and we need to cut them out, okay? If you don't understand anything, tell me, because sometimes my English can be, okay? So here I'm presenting, it's a boring table, from those that had the group, the gross Heliconia habit, okay? So I'm not going to say one by one because it's boring. I'm just going to point some of things that were for me that I need to show to you better. So the medium to tall Heliconia clamp, it was between 1 and 3 meters high, okay? Heliconia magnifica that I planted in 2012 did not reach higher values due to the shelter, because she was reaching the roof and she didn't have more space to go higher, okay? All the others in the clamp area, okay, it was the maximum 2 square meters. And I had a problem with Orthotricha she, because she presented the smallest area, because she was planted outside. And I noticed that she didn't like to be outside in the open field. The sun too strong. So lately, after this essay, or even after 20, I put her inside the shade, and I managed to get at least 2 flowers, okay? So there are other conditions that I have to improve for her to give flower, okay? So I noticed there were other genotypes that grew faster than others, okay, with the shoot emission. So Tropic and Heliconia rostrata after 3 years, and Richmond Red and Heliconia shiadiana after 4 years. So you could see how they look like. In our winter spring, they are like this, because of the cold nights, because our temperature can go until 10, it depends. We can have some hours at 10 degrees, okay? And after you can reach, in winter time, maybe 18 degrees maximum. So they are looking like that. And, even during winter, we have a lot of wind and the leaves are shredded already. And even the sun in winter time and spring, it's too strong for this variety. So they are a little burned in the leaves. So in summer, autumn, I think I did it opposite. This is mixed, I think. Just split it. It's, I don't know, sorry, okay. Because they are more green during summer and autumn time. No, it's okay, winter, spring, it's okay. No, it's not. Oh, I mixed, sorry. I have to check. Okay, but I think it's okay. Yes, it's okay, sorry. Okay. So now we are going for the result of the production, the annual production of inflorescence by Tlinger. Rosa, okay. Look, I don't know if you notice, what I consider as number of plants is usually when I'm planting Heliconia, I'm putting 2 rhizomes in the same spot, okay? So I consider 2 of them, because we're never planting one alone in rhizome. So even me and the growers were planting like that. So Rosa, we just had one plant, 2 rhizomes when we started. But she grew really very fast. At least she spread it very fast. So this is to see that in the second year after the plantation, I achieved the maximum with Rosa. And maybe you will ask why. This is because of the irrigation that I had there. I don't have a tropical climate that I have a lot of rain. No, I don't have it. So I just have a irrigation system. And this one was badly done because it was drip irrigation. It was not sparkling like I wanted, like those we put it for Anturium. Sprinkling. Yes, I didn't have that one. I didn't manage. Sometimes we had problems with money in the center. And so the plants are not developing to the place where it's dry. So maybe I could stay the plant for more years, giving higher yields if I have a better water system, okay? So, but these are the results. I think you can, it's like, it's an approach, for the growers. So As you can see, tulip black was more productive one, but you know, I needed really to improve the irrigation system for that one. So now I divided now the Heliconia because there were 20. Those I put the shortest one. It's mainly the Psittacoro. I think there is a mixture here, Psittacoro with something, but these 4, okay? So Dwarf Jamaican was the most productive, as you can see. For us, she's very good because she's producing in one time that we don't have almost the other ones, and it's the reddish color is for us is very good for Christmas time, and we have it in winter time when we lack flowers with this color. So you can see that some of them, to reach the highest yield, we have to wait 4 years, others 5 years, others are faster and getting the higher production after 3 years, okay? The least productive, it was Nicariensis. But, you know, I'm a little doubt, maybe she could give more flowers because she was growing nearby the wall, and nearby the wall you don't have so much light. So maybe many of them aborted and I didn't manage to get the flower, okay? So I'm not going to read all these. These are from the annual inflorescence of the medium to tall, because in the first slide, as you can see here down, not in the first one that I showed the big table, I more or less classified the genotypes of Heliconia according to their age, okay? So that's why I'm dividing here. So as you can see on the orange color, this is when they reach the higher yield, okay? So from these ones, only 9 of the 16 genotypes with annual yields above 10 inflorescences per clump, okay? Because we need to put something, the growers need some genotypes that will give a lot of flowers, because they have to have some income with it, you know, not one plant that will only give 1 flower per year, as you understand. And the most productive from this medium tall Heliconia were long lover with 24, almost 25, Heliconia rostrata, because this one is an old plant that we had it there before those that we ordered from Costa Rica. This one, the people that immigrated and came back to Madeira Island brought it with them. So Heliconia rostrata and Heliconia lobster claw, it was already in the island when I arrived there 30 years ago. So they're already having this plant nearby the banana plantation, because people from Madeira love flowers. So they usually in their gardens are putting all kinds of flowers they can find. So even in the plantations of banana, they are buying these rhizomes in our center just to put it nearby the banana fields because they like flowers. Okay. So after Richmond Red, okay, and after Tropique. Heliconia magnifica, I planted in the shelter, and also 2 years after, also in the open field, but after you'll see the results. But they didn't flower, okay? So Heliconia she, or Totrica she, I think she didn't develop well. She was always on that size, okay? Very short one. And because I saw, come on, she's not developing at all like this. So let's try to put her in a different place, okay? So I put it in the shelter, and on the shelter I managed to get flower, but not a lot of flowers. I just got 2 flowers. That I said to Russell, but to you. But I think I have to give more light to her. So I'm still learning the best place where to put her, you know, for her to produce more flowers. And Tropique, of course, a lot. So Heliconia magnifica, what happened? She didn't, I don't know if it was the lower temperature that we are getting in February months, you know, or maybe she wanted some shade, more shade than she had it there, where I put it. So 2 years ago, I decided to put her in a different place of the shelter. So instead of being in a corner, I put it in the in the front to the sea with more light. And 2 weeks before I came, all of the clumps that I planted had flowers hanging, you know? So I managed to find the right spot for Magnifica. So we just there I need only to try, you know, different kinds of shading if she's not growing well or producing well in the open field. So this Rostrata, what I wanted to say is, in the third year, she immediately reduced the number of flowers, okay? And I didn't replant, I didn't know why after, again, 2 years, in the 5th year, she got again more. So on that year, on the third year after the plantation, we really got strong winds, okay? So the plants, you see, they were really smashed, all the stems. So for me, the reason why we didn't have, you know, a yield growing up, it was because of that, okay? And you see, we are near by the sea, it's 10 meters, it's very near by the sea. You know, I was expecting that Diana was not so affected by the climatic conditions, you know, because she didn't flower on the 5th year, and after she returned again, but not with so high production. And also I'm going to attribute this to the strong winds and also for of the sea salts that we got it on that year, those years in this months of February, March. That bended and burned also because the sun, even though the temperatures can go a little lower, you know, all of you know that Diana can resist to lower temperatures, okay? But I think it was really that bad year of winds and sea salts that destroyed, and that's why the production went down on that in the 5th year. About Tropic, we had a problem also because she started to reduce after the 4th year. And she's very fast in growing. I didn't know about it. So when I planted this plant, I didn't give the spacing that I gave to the other ones, that usually it was at least on the line 1.5 meters, and between the lines between 2, depends on the area that I had available, 2 and okay, 2.5 meters apart. So the problem, it means that I needed to replant again, to renew the plantation for getting again a higher yield. So I have to hurry. So what I notice is the frequency of crop renewal. I need to make that to maintain an acceptable yield. It depended on the growth rate and the growth habit of the clamp. And to maintain an acceptable yield of the torch ginger and the Heliconia genotypes, the crop renewal is needed after 3 to 4 years for those that have open growth genotypes, and 3 to 5 years for those that have grouped growth genotypes. So these are now the flowering patterns of the torch ginger and the Heliconia genotypes. Okay, what is more reddish? It's the when they are producing more, as you can see, from 1 to 2 inflorescence. And when it is middle color, it's, you know, it's what I said, and the high one, it's more than 2 inflorescences per clump. I'm nervous because I'm running out of time, okay? So for us, you see, our production months is mainly July to August, but you can have it from April to November. So on those, I think I have to go faster, or you'll see later because we'll not have time. So this is important, these patterns. Okay, so that information that I gathered from my observations and what I recorded on the growth habit and the rate of shoot emission of each genotype is important to define the plant spacing. And after, to maintain an acceptable yield, the frequency of crop renewal must be evaluated for each genotype because it depends on plant spacing, on the habit and growth rate. So for the growers, the different flower production and flowering periods among genotypes can help growers to decide which ones to choose. And for them, it is economically advantageous to choose those that have those genotypes that ensure an extended flowering period, because our clients want constant supply regardless of the amount. So because of that, I made some files for them to see that I'm distributing to them, to those that are interested. So I put it, the torch changes, where I can put the annual yield that I got it. I think they'll manage to get it higher with better irrigation and fertilization and things like that. The flowering, when they are flowering, when they have to renew the plantation according to the spacing that they are using. And the campaign, that's also important because of the winds, okay? So I'm just going to pass, this is the Heliconias with open growth. That one, the Golden Torch and Guyana. These are those with grouped growth, more with upright and some that you see. That photo is from the now, from the 2 months ago, 1 month ago. Okay, the Collinsiana is not producing so much, so they are not going for that one, I'm sure, our growers. So this, the Rostrata. And I think I have 2 minutes. Okay, Wagneriana, she's flowering every year now inside the shelter. I loved it, that one. So thank you. If you want, so you can give me your email and I can send you my presentation with the tables, attached with the tables of the spacing that I use, because I think it's important. And also because I can, not this, I can send you the data from the Meteorological Center that we have there, and also the spacing that I used, okay, because it's important. Okay, these 2 tables, okay? Thank you very much.
So next, I want to invite up Dr. Pascal Atalor. He is stepping in for Dr. Dong Cha. Thanks to our new administration, United States Department of Agriculture is suffering a lot of financial difficulties, including funding freezes. This means that a lot of these researchers can't travel because travel funds are frozen. Dr. Cha was going to come and give the talk, but we're really lucky to have Pascal here. Just a little bit about each of them. Dr. Shaw is a research biologist at the US PBARC, that's the Pacific Basin Agricultural Research Center in Hilo, Hawaii. He is a neighbor of Pascal and I. Pascal himself is now entomology specialist, so it's arthropod monitoring and identification, but he's also doing a lot of integrated pest management work. Pascal and I work closely together because he's focused on the floriculture and nursery industries statewide. So I would like to invite Pascal up, and I'll give you a chance to give a little talk while I get your slides up there. And I can remember the lays about 50% of the time.
So my name is Pascal Atalor, and my talk today on behalf of Dr. Dong Cha, as Russell said, at USDA. Sorry, can you hear me now? Okay, so I was saying my name is Pascal Atalor. Giving this talk today on behalf of Dr. Dong Cha. As Russell said, Dr. Dong Cha is the research chemist at the USDA.
You got a presenter view right here as well. Check to make sure the clicker works.
Okay, so my talk today on behalf of Dr. Cha is a systems approach for ornamental crop exports in Hawaii. So as we all know, globalization facilitates the rapid and easy movements of people and goods. But unfortunately, it is also a significant driver for the spread of invasive and phytosanitary species that cause trade barriers. So our goal is to develop control systems to remove trade barriers by having pest-free plants. And why is this important? The ornamental industry in Hawaii has declined in recent years. And this is because, for example, if you look at this, you see that in floriculture alone, there was a 7% drop in export sales in 2023. That's very worrisome. And one of the challenges or causative factors for the decline in the industry is the stringent phytosanitary standards that results in the rejections of nursery plants and cut flowers. So our goal is to develop effective treatments to reduce rejections for the industry. So we conducted a systems approach study, thankfully with funding from the Hawaii Floriculture and Nursery Association. Okay, so the traditional or conventional practice to achieve protein security usually involves a direct standalone post-harvest treatment to obtain a probing 9 level control. However, this usually involves treatments with high intensities, such as boiling. Of course, boiling will kill insects, but it can also cause phytotoxic damage to plants. So hence the importance of systems approach, which incorporates multiple or several pre and post-harvest components that reduce pest risk at each step or component. And thankfully for the ornamental industry in Hawaii, Dr. Arnold Hara of blessed memory tested several, developed and tested several components of the systems approach. So we tested if the combinations of these components will be effective against the different target insects. So the question now is, what pests are the main culprits of rejection? And to answer that question, we chronologically summarize a 5-year historical rejection data. And the following pests were identified. However, 6 groups of these pests were responsible for 80% of the rejection. This means that if we effectively control those 6 groups, we can successfully reduce rejections by 80%. So here, the flowers with the highest rejection rates were determined as ginger, Heliconia, and dendrobium. And the pests associated with them includes millibugs, scale insects, aphids, ants, thrips, and sloth. Now, looking at these beautiful flowers, they appear to be very clean and devoid of pest infestations, right? But when you zoom closer, you have a bunch of insect infestations in them, hence the importance for control. So like I said, Dr. Arnold Hara tested several insecticides and hot water washing treatments for ginger and heliconia. So we tested if the combinations of these components would be effective to get pink plants and to quantify the effects of each component. We surveyed the flowers for pests before and after each step. So at post-harvest, almost all the flowers were infested. And these were the insects associated with the infestations. However, after the first wash, you can clearly see significant reductions in infestation levels, as well as in the proportion of infested flowers. After the second wash and hot water, even more reductions, as well as reductions in the proportion of infested flowers. So the system works, but unfortunately not well enough because these flowers will be rejected if inspected. So in 2018, we repeated the experiment, excuse me. But this time with the inclusion of insecticides, and specifically marathon, safety, and control, but still no complete control was obtained. However, in 2019, we replaced marathon with contest, and complete control was obtained. So the system works, and but quality control is super important because if each step is missed, it can result in infestation. Okay, so the industry wants a last step insurance for this system, and that is fumigation, which is the next part of my presentation. Okay, so one of the fumigants that we tested was ethyl-formate. Ethyl-formate is a natural product that is generally recognized as safe by the FDA. And there are two products available. One is a cylinder pressurized with carbon dioxide, and the second is a vaporizer with liquid ethyl-formate and nitrogen gas as propellant. Okay, so for ginger, we tested citrus mealybug because it is one of the most pesticide-resistant mealybug species. And so one of the treatments we tested was a high-dose ethyl-formate for 4 hours, specifically at 70 grams per cubic meter. This treatment is known to be effective against the pest, but it can also cause phytotoxic damage to flowers. So the second treatment we tested was phosphine for 24 hours. The exposure of phosphine for 24 hours is effective, but 24 hours is not expeditious for sugar. And the third and final treatment we tested was the combination of a low-dose ethyl-formate and phosphine for 4 hours. So the difference between the combination of ethyl-formate and phosphine is that here, the exposure time will be for 4 hours, but with phosphine alone, it was for 24 hours. So here shows that we conducted the experiments, and also very importantly, we did assessments or measured for phytotoxic damage on the flowers, so measuring the chlorophyll content, and hue index, and the wilting index as well. So the high dose ethyl-formate and phosphine for 24 hours cause complete control of the citrus mealybug. However, the phosphine for 4 hours and the low dose ethyl-formate were less effective. But the combination of the low dose ethyl-formate and phosphine for 4 hours also cost effective or complete control. And here is the most important of the results that we got. So if you look at that, the high dose ethyl-formate, you can clearly see phytotoxic damage of the flowers there. But for the combination of the low dose ethyl-formate and phosphine for 4 hours, there was no phytotoxic damage. So this combination of the low dose ethyl-formate and phosphine for 4 hours is currently being optimized for export of ginger in Hawaii. So here is just more quality control data showing that the combination of the low dose ethyl-formate and phosphine for 4 hours was the most effective treatment without any phytotoxic damage to flowers based on the hue value, chlorophyll content, and wilting index. Okay, so this is another important export flower in Hawaii. This is the white dendrobium. And unfortunately, thrips are one of the most important phytosanitary pests of the white dendrobium. And as you can see here, despite the different pesticides and the durations that we tested, none of these insecticides were effective because thrips are notorious when it comes to evolving resistance to pesticides. Okay? So we did some fumigation tests using a thiophomate, and you can see it was very effective based on the lethal concentrations that killed 50% and 99% of the populations of the western flower thrips, the flower thrips, and thrips palm. So this was a setup for the fumigation test of having the treatment on the right and then the control on the left. And also importantly, we measured some potential effects of the treatment on flower quality and base life. And here you can see that after 7 days of treatment, the quality of the flower, the treated flower, was essentially the same as the control, indicating that ethylphomate is a potentially effective post-harvest treatment for the dendrobium. So these are just other projects that are currently running in Dr. Charles' lab. And I'm also closely collaborating with him for the fumigation test for the coconut garden cirrus beetle. And Dr. Chow would like to thank his postdocs as well as all the funding agencies that support his work. And thank you very much. Any questions?
The hot water, what's the temperature that you use that hot water?
42 to 50 degrees Celsius. Yes.
What about the cost of these treatments? How does that impact the profits that the growers will take? I mean, obviously, they'll get more than if the shipments were rejected.
Yes, so of course, initially it's going to be very expensive to set up, but on the long run, it will be cost effective because, for example, if you use insecticides alone as a standalone control, it may be effective now, but it has, especially thrips, will evolve resistance over time. So you just keep spending more money buying different insecticides that would not work.
Just repeat the question if you did it.
Oh, okay.
I have 2 questions. First one's very quick. What was the brand name of that phosphine you guys are using? Or do you know?
Yeah, so these are not registered yet in the United States.
Okay, cool.
Watch what you say. He works for HDOA.
I'm curious. The one phosphine doing that I do know of is a restricted use pesticide.
Yeah.
So that would really make it difficult for local.
Thank you for your trick question, man.
My follow-up question.
Yes.
Have you noticed in your discussions with the horticulture industry any hesitancy to engage with these new types of chemical treatments aside from cost? Is there a concern about safety? Is there a concern about compliance? Are people excited about these new options, or do they think like, oh no, pesticide, I won't touch it?
Yeah, so the question is concerns about safety and also compliance of insecticides in the industry. I just completed a pest needs assessment survey for the industry, and with that, I'm currently analyzing the data. So right now, I cannot answer that question until I have completed that.
Awesome. I'd love to talk with you a little bit more after.
I was just wondering if you use in any planting or, elimination of pests instead of the insecticides and the other irrigation methods that you use. Just a obvious gardener, but I do use herbs and, like the flies, the aliens, even some of those, some, other herbs, or different marigolds and things like that, which actually have been hugely successful, just getting rid of common garden, snails, slugs, aphids, yeah. So I don't know whether you do that, or whether that's a possibility.
Right. So the question is to summarize, like the role of companion plants in helping or assisting pest control. I'm not sure the industry does that, but I think it's important because companion plants are important because of the potential of releasing specific volatiles that can either repel or attract different pest complex. So I think it's an important aspect that the industry needs to actually look into. Yeah.
One other question. So after heat treatment, Pascal, does the cut flower remain the same as fresh as it is when it's freshly cut? So I think that was a little bit about the wilting potential. And
so it depends on the temperature.
Okay.
So the temperature is very important. If you set it up too high, of course, it's gonna have issue.
In Hawaii, you remove the flowers from inside the bracts because the big Heliconia, like the eye, we have a kind of spoon, something like this, that we hold the bract and remove all the flowers because this is a place that have a lot of insects.
Yes.
And the year you remove because as you show us, scales in leaves, but we don't sell the leaves. We just use the flowers. Here they remove the flowers from the inside the bracts, no?
I don't think so, but the problem is, but even if the insect is on the flower or wherever part of the plant, if that plant is inspected, it's going to be rejected because of the presence of just one insect.
Yeah.
Yes.
So we've got a great group this morning that's going to be doing a couple talks and a short workshop for us from our very own Tropical Plant and Soil Sciences Department here at the College of Tropical Agriculture and Human Resilience. Recently name changed. Da, 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 da. We got 3 presenters, 2 of which were advisors for me during my graduate program. And so I'm very happy to have them here. First, we've got Dr. Ted Radovich, who at Tropical Plant and Soil Sciences Department is a specialist and professor in the department that I just said. He focuses on optimizing crop yield and quality in sustainable agriculture systems. We also have Dr. Amjad Ahmad, who is an associate extension agent here in the same department, and he focuses on sustainable ag. With them, we also have Elima Ho Lastimosa, who is a prominent Native Hawaiian educator and community coordinator at the same campus that we work at here. So we would like to invite them up and thank you to all 3 of you. And just so you know, Ted, the mic is super, like hold it.
Okay. Thank you, Bob. Appreciate it. Yeah, thank you. Is my thing up here? Aloha, gang. Welcome. Okay, so we were asked to do this workshop. So I'm going to go ahead and get it started and talk a little bit about turmeric, primarily Curcuma longa, but some of its relatives in the same genus. Dr. Ahmad will come up and talk a little bit about ginger and some of the work that he's done. And then Auntie Lima will have some of our Olena, our turmeric, and others in the back. And we're going to have a little hands-on activity just to get the blood flowing.
Sorry, could you?
I sure can. we get some kupuna in the room, huh? Okay. How's that? Is that all right? Wow, okay. Usually I haven't really been accused of being soft-spoken before. So that's that. So what is my presentation on here?
It's already loaded up.
Okay, I don't see it. Oh, is it up there? Oh, okay. I just don't see it on the screen. Thank you, Gabe.
Oh.
No, I got it. I can do it. I just didn't see it on the screen. No, it's good. We're here. I'll just.
There you go.
Yeah, okay. Mahalo. Okay. All right, so most of us are probably familiar with curcuma longa. It's historically very important. Many years Ayurvedic medicine, traditional Chinese medicine. It is a Polynesian crop as well. It came through across, and it's been here in Hawaii for a very long time, and very important for medicine for sure, used for dyeing materials, a whole host of things. The leaves, for example, are often substituted for tea leaf for use for extra protection in lei and things like that. So it's important for numerous reasons. Curcuma longa, particularly here in Hawaii, as well as most of the continental U.S., really blew up in popularity when there were a series of publications that confirmed curcuminoids as effective anti-inflammatory agents, which was known already in other medicinal traditions. But once it was published in the literature, popularity really expanded, and everybody was looking for sources. And Hawaii, because of its political situation, association with U.S. And certified organic and a bunch of other types of labeling became really high in demand. So there was a big interest in producing this material. The farm gate floats around $5, but retail price can skyrocket. It can be $15 for fresh material in the stores. It usually hovers between 10 and $15 in the store. It could be even more. They'll sell very small amounts for a few bucks, and if you multiply the weight, then it ends up being in the 20s or 30s per pound. I don't know if you can see this, and Dr. Ahmad might have this slide too, but it has turned into one of our highest valued specialty crops. So about 3/4 of the way down, you see turmeric. It's valued at over 2.5 million, which for Hawaii is big. And it's closely followed by noni, which is unrelated medicinal crop. And the other two members in the same family, galangal and ginger root, are less than half of the value now. So it's important. This is a picture of one of our fields out at the Waimanalo Research Station. Can you guys hear me okay? It sounds like I got to put this thing right up.
Yeah.
Kiss this thing. Okay. So this is one of our fields. So in the last several years, we've been focused primarily on genotype selection for curcuma longa. We have a bunch. So we currently have 9 of longa that we've pushed forward that we continuing in our multi-island trials. These 5 were some of our earliest that we worked on. Those 3 on the left, Olena, Hawaiian Red, and BKK, were already here. Puno Organics on the Big Island introduced them. And then we introduced a few more that we've been trialing, including Roma there and Joy. And we'll have some data for you right here. So over the course of 3 years, 2019 to 22, we trialed these on multiple islands, multiple locations. And Roma is a variety from India. We worked with Karnataka Ministry of Agriculture and our Department of Ag. And it's a beast. It produces very high yields and pretty good curcuminoid content. Curcuminoid content is also real important. I'll talk about that in a second. Hawaiian Red, which is not a Hawaiian variety. They just labeled it that. I don't know its history. It probably was collected in Thailand. But Hawaiian red is our industry standard, and so we've found a number of these curcuma longas that yield better for us than Hawaiian red, although we keep Hawaiian red in the rotation because it is a good performer. So curcuminoids, curcumin and then a couple of its analogs are very important in the industry with regard to quality. And so oftentimes our producers will be asked by buyers what the curcuminoid content is. That dotted line there is the lowest level of acceptability, 4 to 6% is kind of the range of average. And so our Roma is, for its yield, is a real good one. We have a new variety that we're not releasing yet because we're working with the community in India where it came from to make sure we do it right. But we have some other material that's even higher and larger that we're pretty excited about. BKK is consistently our highest curcuminoid content. The challenge with BKK is that, let me see if I can go back. Yeah, challenge with BKK, it's also consistently our smallest rhizomes and our lowest yield. And we have a bunch of material on the back that you can take a look at. Okay, this is Roma. Nice big guy. Sitalingi. We'll go back here. The color, the correlation, curcumin is a color compound. The correlation between color and curcuminoid content is significant, but it's not so tight that you can really differentiate above, 4 or 5%. But you can look at, so Indira types, which are the yellow types, we have an Indira type here. You can see it in the middle. Light color is usually indicative of lower curcuminoid content. And so it can be used as a rough screening tool. And Roma, one of the challenges of Roma is that throw sports, sometimes lighter colored sports, kahuli. So we like the Sitalingi. You can see the consistent orange content there. Okay, we have a number of accessions, probably 10 different species. The nomenclature on these guys is pretty muddled. We spent a little bit of time with Jana Skornikova over in Singapore Botanical Gardens, and she gave us a heads up on some of these things. So a lot of these species, I don't know if you're familiar with Xerodaria, there were a couple of species under that name that she teased apart, and now are two separate species, Daria, which we have the true one, and then Picta, which is the one with the red stripe. Curcuma aruginosa is very important in Southeast Asia, Indonesia. It's used in all kinds of preparations. There's a bunch of different colors, probably multiple species under this one name, and so there's a lot of nomenclature work that needs to be done for sure. And then Curcuma caesia, some of these blues, some of these aruginosas are often marketed as caesia, what they call black, which you'll see is just a really, really dark blue. Anthocyanins are expressed pretty, is quite high, but it's not black. But you will often see the aruginosa, blue aruginosa is marketed as caesia, because caesia is popular and gets a higher price. In the same picture. So here are some pictures of flowers. Nobody's in flower right now, so we don't have any fresh flowers. This is one of our aruginosa sessions. We're calling it green. It has concurrent environmental influence is quite strong on the expression of the carotenoids and the anthocyanins. So the green has probably of the aruginosas the highest expression of yellow compounds, and so the co-expression of the blue and the yellow often makes it look like it's green. But the flowers are gorgeous, and it's probably our freest flower. During production, there'll be quite a bit of flowers on it, and you can see the coma's got that nice bright pink on the end. This rubescens, Curcuma rubescens, we're interested in the rhizomes. It produces pretty heavy, large amount of rhizomes. It's used medicinally. Not a lot known outside traditional medicinal circles, but some of these are also used for landscape industry. So ruby ginger, they call it. This is a wild type species, but it has potential for edible landscape, etc. And just want to mahalo the large group of folks that have helped this work and will be available after the top story. And again, we have a lot of that stuff in the back so we can talk some more. But I want to invite Dr. Ahmad, Amjad, you want to come up? Yep.
I'll get loaded up, just do a little talking.
Good morning, everybody. So my presentation is going to focus on ginger today. And the title is Soilless Ginger. And because multiple issues in ginger in Hawaii, including bacterial wilt, which can cause decline into complete elimination of production. So we wanted to focus on soilless ginger, especially for people who can produce the seeds for us in order to make money. That's for them first, and also to increase the availability of ginger, clean materials ginger in the state. So that's why the presentation is focusing on that, and that's why it's benefits and best practices as far as what's available and what's good to do to produce soilless ginger. Ginger is a native to China and India. Sorry, I didn't memorize all this text. To India and China, and zingiber comes from the Sanskrit word of stringa vera, which means the with a body like a horn, which related to the shape of the ginger tubers itself. Ginger has been important in Chinese medicine for many centuries and is mentioned in the writing of Confucius as well. In English pubs and in taverns in the 19th century, barkeepers, that's where the ginger ale actually came from. Barkeepers were to put some ginger next to people they are drinking the beer to add to it, and that's where the ginger ale actually came from. I'm a Muslim, I'm not supposed to say it, but went through already. Ginger is known as a diaphoretic, meaning it's causing one to sweat. Maybe you can tell after drinking some ginger, you see the internal heat comes out as a sweat. And that's why Henry the Eighth actually recommended the mayor of London to include ginger for that benefits, thinking that the sweat will help also the plague to come out of people because of that sweat that's happening after drinking some ginger. Especially if you drink the shots, the concentrated ones, you see it's like coming out from the head. Yes, it's very clear effect and immediate. You drink from here and then you feel the heat, the internal heat comes as sweat immediately after. Ginger is a carminative, means that it helps in the breakup of intestinal gas. And that's why Indians, they use it a lot in their cooking because they eat lots of beans, which causes gases. They add lots of ginger in there to balance that gas issues. Additionally, it has been used remedy for congestion and as a compress either on the sinus or on the chest, and it's often used as a combination with other remedies for general tonic or stimulant purposes, or to prevent gripping as well. Ted mentioned this table already. Dr. Radovich already mentioned that table. Ginger is not on top as far as the production and the consumption, but actually has been increasing. And you can tell from the last column, where there is a 400 additional acres that's been added in ginger production. And you can tell that there is definitely an increased demand, and that's been helping to increase the production or driving the production additionally in Hawaii as well. Now, Hawaii Island, the big island, is the biggest producer for ginger in Hawaii. However, if you talk to people who kind of been in Hawaii for a very long time, they tell you that the farmers used to completely stop producing ginger in certain areas because of the bacterial wilt. They will completely abandon certain fields and go into new fields. And then until the bacterial wilt becomes so dominant there, then they move into another field and another field over time. So additionally, with also moving with that, been also increasing the bacterial wilt infection and also viruses. Because especially between the islands, we can move seeds and propagation materials as long as not with soil, no issues. And that's been an increasing of distribution of the bacterial wilt itself statewide, and also some viruses in the state too. So there are definitely challenges with the bacterial wilt and the viruses spreading out. Looking for clean materials will be the best way to combat that. Bacterial wilt, the symptoms of it looks like senescing in ginger. Senescing is the physiological changes when the ginger tells you, I am ready, come harvest me. But when it happens early in the growing season, that's usually a bacterial wilt, where it starts with wilting, vascular discoloration, if you make cuts in the plants or even in the stem, and additional eventual plant death. It can cause 60 to a complete elimination of the crop completely. You don't harvest anything out of it. So that's where it comes the soilless ginger. We wanted to focus on producing clean materials. That's one. Second, expanding the production without taking more of the land. We have limited lands, but then on the big island, we have huge areas of land that's all lava rocks that can be easily used for soilless production of any kind of crops, including the ginger. Additionally, if farmers will produce clean materials, they will make better money selling those clean materials for production as well. And also the same ginger can be used as a consumption too. Now, in order to expand that, we wanted to look into what's the condition, make sure there is a clear procedure for people to adopt. For example, slopey areas, rocky and lava land where you cannot plow, and arable lands. All that areas can be used easily. The edges of a farm, for example, that can be used as well. The only thing what you need, what you need is benches, and then put the pots on top, and you can reuse the same materials over and over. We wanted to make sure that farmers, no matter what's their operation, things are ready for them, and we tell them what's good to use. For example, organic farmers or conventional farmers. We looked into different materials, synthetic fertilizers, slow release types, or organic fertilizers as well. You can tell usually based on the trials that we did that if you use organic amendments, you don't have any kind of deficiencies in the growth, because usually organic materials, they will provide the plants with all the nutrients at various amounts. When it comes with the synthetic, they are only NPK, except when it comes to NutriCoat and Osmocoat. When you use them in the NutriCoat, there is additional more nutrient, and that's based on our studies that they are more beneficial. When it comes to the media, for example, the pots can be plastic or fabric. There is no difference in the use of them. However, what we found that the diameter of the pot can impact the yield. Same 15-gallon pot. One is bigger diameter, a bit shorter. The other one is taller with a smaller diameter. I had a significant difference in the yield. I harvested from one of them 11 pounds of ginger, and from the other one 15 pounds of ginger. So there was a significant difference, just the same volume, different diameter. When it comes to the media, coconut coir or peat moss, both can be good. However, over time, the peat moss will compact and cause some tuber rots, while coconut coir stays more porous all the time and allows the water to flow through, and there is much less incidence of tuber rots comparing with to use peat moss. Varieties, Hawaiian, Jamaican, and Peruvian ginger, we tested the three of them. They are all did very good as far as ground and as far as growing them in pots. Definitely, they have some difference in characteristics between them. And definitely the Hawaiian is the best growing and yielding variety in pots and in the ground comparing with the other varieties. When it comes to the setup of the soilless ginger, we went through all the procedure to make sure people who adopt the practices know exactly what they should do. So as far as the setup, how much is added of the media over time, and also the fertilizer itself. Like for example, if you are using synthetic fertilizer, we recommend only 4 applications from the month 2 of the growing season. When it comes to the organic amendments, we recommend 5 applications because usually organic amendments produce some heat when they are breaking down, and that could cause some issues with the, for example, burning of the edges of the leaves due to that heat that comes from the organic amendments. We made a complete schedule for people to apply if you are using synthetic or if you are using organic, and different combinations to make sure the crops are getting the optimum amount of nutrient that they need for optimum yield as well. And then also, again, as I mentioned, the applications, if it is from synthetic or from organic amendments, when and how many times that amount of fertilizers has been added. Harvest, it's very easy. You just empty that pot, take the media, and it's a very good system because you can take all that media, compost it, and you can reuse it back again. The pot can be reused, the media can be reused after being composted, and then the same ginger also, you can take some of the ginger that you are harvested to start over. In Hawaii, we plant ginger between March and May. So technically right now we start kind of planting till about May, and you harvest between January and March the year after. So after you harvest, you can immediately start your planting the second time after with the same materials that you harvested from. Growth, definitely the yield increased with the increasing of the pot size. We went from 7 to 10 to 15 gallon pot, and the yield went up and up, responded to the size of the pot as well. And here is a recipe for you for pickled ginger. You can snap a shot of it if you want to. It's very easy, very simple, and very tasty. In Hawaii, we say ono. That's tasty. Thank you so much.
There we go. So I've been working on this project for about 5 years. We thought we were going to get it knocked out pretty quickly. Turns out, like most things, taxonomically and otherwise, it takes a long time to solve some big issues. So if you've been in Hawaii or you went to the Columbia conference, you may remember my talk about this a couple years ago, or every year that you've seen me at a research seminar in Hawaii since we started. Just a little background. I don't want to go too into the disease complex itself because we've covered this a lot in other groups, and I want you to be able to understand where we're at right now. Since 2014, red ginger, Alpinia purpurata, you can see it's kind of a poster child plant for Hawaii, seen often in most flower arrangements and a lot of media. It's a very common and popular flowering plant for use in Hawaii floriculture and otherwise outside of Hawaii. Production began to decline before 2014, but growers expressed extreme concern around 2014 that an unknown disease was causing complete collapse of red ginger stands. The disease started on Oahu, but it has since been identified statewide. Floriculture production and plant health has been severely affected by this, with some growers going out of business. We've lost over a third of our growers since 2014 that were growing red ginger because of viral issues and most likely some other conditions. But the virus is expected to be a big portion of that. So viruses were identified as the causal agent. We went through bacterial assays, we looked at fungal pathogens, and we also did a lot of virology work and determined that most likely the viral pathogens that we found were the causal agents. That work happened from 2018 to 2021. The symptoms you're seeing up on this slide are what growers have been experiencing. When you step back and look at the general farm symptoms, you're seeing stunting, dieback of plants. So normally plants are closer to 6 to 10 foot tall. We were seeing plants that were closer to 1 to 2 feet. They also had necrotic growth, complete dieback of the rhizomes and the stems and leaves, to the point that some stands completely collapsed. We said viral pathogens had been identified. This table is showing you what those viral pathogens are. So on the left-hand column, you've got the virus itself, candy yellow mottle virus, banana bract mosaic virus, bean common mosaic virus, banana streak virus, and 2 new viruses that we identified that were never found outside of gingers, outside of Hawaii. So we identified 2 new viruses, Alpinia vein streaking virus and Alpinia vein clearing virus. The vectors for these viruses are varied. So we've got mealybugs, which most likely vector candy yellow mottle virus. We have identified them as a vector, but there may be other vectors as well. We do know pretty well that aphids vector banana bract mosaic virus. Aphids possibly vector bean common mosaic virus, possibly mealybugs vector banana streak virus. We're not entirely sure if they're the only vector or if they are the primary vector. And then we did identify that both mealybug and aphid vector Alpinia vein streaking virus and Alpinia vein clearing virus relatively. We do know how well these vectors vector the viruses. We did transmission studies to see how efficient these insects are at vectoring these 3 important viruses. So I've got 3 viruses that are in bold because you can see on the penultimate right-hand column the total number of incidents. It's showing you how often we found these viruses, right? Banana bract mosaic virus, bean common mosaic virus, or bean common mosaic virus and banana streak virus. We didn't see it a lot, right? Out of 100 samples, we only saw BBRMV, I'm going to start shortening them, 18 times. BCMV, we only saw 3 times. And banana streak virus, we only saw 5 times. So we sort of threw these to the side. We don't see them too much. We're not going to worry about them from a production standpoint, right? They're not as big of an issue for growers because we're rarely seeing them. So we're really focusing on trying to reduce and understand and eliminate candy yellow mottle virus and the 2 new Alpinia vein streaking and vein clearing viruses. Just going to give you an idea of what some of these look like. So we've got BBRMV, banana bract mosaic virus, called mosaic virus because it causes these mosaic patterns. We've also seen symptoms of it causing flower breaking. Some folks think this is really attractive, so they are cultivating them more. I also think they're attractive, but I want to elucidate that it is a virus that's causing the color breaking. Candle yellow mottle virus generally causes symptoms either just vein streaking or vein clearing, like on the right-hand side, but when it is severe or more severe, it causes mottling and vein clearing. It gets really bad when you start to have multiple viruses infecting the same plant. And that's where we're seeing a lot of issues for growers. This is a plant that has been infected by banana bract mosaic virus and canada yellow mottle virus. Once we see 2 or more viruses infecting one plant, that's when we start to see pretty severe decline symptoms in individual plants or whole stands. Here's another example of multiple viruses infecting a plant. The symptoms start to become very complex, but you almost always will see color breaking, mottling or mosaic, and of course stunting and decline in the plant. One of the viruses that's difficult to identify visually, but still affects plant is the Alpinia vein streaking virus. You can see a little bit of the vein streaking in that image right there. But for the most part, Alpinia vein streaking virus is very hard to detect unless the plants are in a condition where they're very unhealthy. On the left-hand side, you're seeing a little bit more of that vein clearing. You can see that almost variegated look, right? You seeing what I'm saying? Yeah. Okay. But the picture on the right is trying to exemplify that you sometimes only see it in, for example, plants that are receiving too much sunlight versus plants that are in shady conditions. So this was at growers where we went, and only the plants on the left-hand side were showing vein streaking. The plants on the right that had shade had no vein streaking. All of them tested positive for Alpinia vein streaking virus. So plant health, it plays a role in virus expression and ultimately plant health and yield for those folks that are worried about yield. So when we see 2 or more viruses in a plant, this is really what you're starting to see, right? So these 2 plants tested positive for both Alpinia vein streaking virus. The one on the left also tested positive for Alpinia vein clearing virus and canny yellow mottle virus. We used to call it virus B because we don't have a name for it yet. So I didn't update that slide. Whereas the picture on the left, it's just Alpinia vein streaking virus and canny yellow mottle virus that are infecting these plants. But you can see once you hit 2 or more viruses, you see plants that essentially are unproductive, won't be nice plants for either a floriculture producer or somebody in a landscape. Alpinia vein clearing virus also causes really unusual typical vein clearing, but bleached and distorted foliage. We were having a really hard time figuring out at this one growers whether or not they were experiencing herbicide toxicity or virus. So we actually grew these plants outside of an herbicide affected environment, and we still got bleached and distorted foliage. And it can get really bad. You can see this all across whole stands of plants. And eventually, with just Alpinia vein clearing virus as the only virus infecting a plant, you can have complete stunting and loss of production. We've also seen it in some other plants, although it's almost entirely been found in Alpinia. I have yet to find it in Heliconia, despite testing several plants. We did identify banana bract mosaic virus in one Etlingera here on Oahu, and then I just saw this beehive ginger at Lion Arboretum, so I want to go get a sample from Lion Arboretum. Orville actually pointed it out, so thank you, Orville. We might have a new plant that's being affected by, I don't know, maybe candy yellow mottle virus. We'll see. But general conclusions, I'm not done. General conclusions are that all these viruses can cause decline in the plants, but really Alpinia vein streaking and Alpinia vein clearing virus are the most commonly found plants. They can both cause complete decline in the plants and in the stand. And then once you mix them with another virus, you are nearly 100% guaranteed to have plant health issues to a degree that would make them unsuitable for neither floriculture production nor landscape use. These have almost guaranteed been spread primarily by vegetative propagation. One reason I say that is the vector efficacy studies that we looked at. And when you're looking at 8 to 10% efficacy in transmission, more than likely, the insects aren't transmitting these viruses enough to the point that they would be this widespread. To see almost to literally no virus-free plants in the state or outside of the state. So you got to start with virus-free plants, and that's where we've been at for the last 2 years, is finding virus-free plants that we can multiply and then start to release to the industry. And once we had issues there, making virus-free plants through tissue culture practices. So we're looking at tissue culture to create or to multiply currently known virus-free plants. Two methods that we've been using have been meristem isolation and thermotherapy. We haven't looked at chemotherapy just because neither of the tissue culture labs that I've collaborated with have been interested in using chemotherapy. So you essentially will collect the shoot tips, submit them to temperature extremes that would kill off the virus but not kill off the plant, or try to isolate the meristem to the point that you do not have tissue with virus load in it and then grow with that virus-free tissue. About 3 years ago, we thought we identified virus-free plants. They originally tested positive, so we collected plenty of these plants, made an insect exclusion cage inside of an insect exclusion greenhouse, grew them in this cage before we tested them again 2 years later. Turns out they were virus. So we could not use these plants. We had to move forward with finding other virus-free plants or putting these plants into tissue culture, which we have started about a year ago. I scoured the globe, maybe not the globe, just America, for tissue culture sources of Alpinia purpurata. I put together sources and ordered from those sources, one of them being AgriStarts in Florida. We bought red and pink ginger from AgriStarts, brought them back. They looked pretty good and tested them for virus and only found 2 red ginger in the 74 plants that we bought to be virus-free. So you can see that red 1 and red 10 have no virus in them, right? We're not positive that they have 0 virus because look at the rest of them. They all have either banana bract mosaic virus, bean common mosaic virus, or possibly Alpinia vein streaking virus. So we found Alpinia vein streaking virus somewhere else in Florida.
Did you let them know that so many that they delivered had the virus?
Yes. Whether they let you know, I don't know. Where did AgriStarts get their tissue? I don't know. I don't know. I'm not entirely sure. I haven't tested any plants outside of Hawaii other than these. I would love to test some plants elsewhere because it'd be really interesting to know where these viruses came from. Alpinia is not from Hawaii, as you all are probably aware. And so somebody brought it in, and more than likely, somebody brought it in with at least some of these viruses. The fact that we found Alpinia vein streaking virus and Alpinia vein clearing virus, and they were never described to science before, indicates that it's possible they're in other plants. We just don't know yet. It would be interesting to test some of the other plants that share the other viruses, banana, canna, some Indozinda borealis. But so far, we haven't found them in other plants because we haven't looked. We took those 2 plants that were virus-free and we've thrown them into tissue culture. I want to thank Elisa Mao. She's our tissue culture lab manager at Komohana Research Extension Center. Somebody tell me how my time is too if I start to get close because I'm not checking myself. We're good. So we have started with initiation and multiplication of the 2 virus-free plants, mother plants. Now, I'm growing out those plants in a virus-free environment because we want to make sure that AgriStarts gave us the right plants. We want to see if it's a form of Alpinia purpurata that's interesting and useful for the floriculture industry. You might know Alpinia purpurata sometimes throws up a lot of the actual flowers through the bracts. And the floriculture industry does not want, thanks, Eric. Floriculture industry does not want to have a lot of those flowers because of what Pascal was describing yesterday. We have issues when there's more flowers inside of the bracts because it's a lot harder both to clean the plants to be ready and presentable to growers, but also to clean the plants and have them free of insect pests, because when you have those flowers down in there, it's just easier to hide. So Lisa's working hard at initiation and multiplication of the currently known virus-free plants. She's also working on meristem isolation to remove virus from some of the plants that we know we want, Kimi, the Jungle King, and the Alpinia purpurata forms that we're familiar with here in Hawaii and we want to produce. We have a protocol that we've been using. If you're interested in it, I'll just briefly go over it, especially because this isn't a tissue culture crowd, but it's already published. The publication is above: efficient regeneration of Alpinia purpurata plantlets from rhizome bud explants. And they've got a full protocol, and we didn't even have to create our own protocol. We just ran it off of this, and Elisa was able to successfully initiate and multiply the plants using this protocol. We've also been working with collaborators at the United States Department of Agriculture's Pacific Ag Basin Research Center. So that is Dr. John Suzuki and Dr. Tracy Matsumoto. You see on the right-hand side is a representation of how our relationship has been working. I get plant material and I help them find plant material from either local sources or external sources off island, like AgriStarts. Also, Tracy and John are finding plant material. John is now doing all the diagnostics. He's doing the testing for us. So when we send plants to check them for virus, we're sending them to John Suzuki. It's great. He's right across the street, so I don't necessarily walk them over, but he's somebody that's readily available. We also put those plants into the greenhouse to continue to grow them. He then gives plants to Tracy, and Tracy manages the tissue culture lab at USDA's Peabody Center. Also, I want to thank Brienne Corpus, who is John's lab tech. She's doing most of the actual virus diagnostic and John's managing the program. Tracy is also working on initiation and multiplication of the 2 virus-free plants that we found from AgriStarts. We need to test them again, of course, so no guarantees that they're fully virus-free. She's also working on meristem tip culture and thermotherapy to eliminate viruses from these plants. And then ultimately, we go back and forth through virus diagnostics to attempts to eliminate the virus until we get virus-free plants that we then multiply and release to the industry. Tracy has successfully eliminated virus based on initial diagnostics from 2 plants that have been put in that were originally virus infected, but are now testing positive, negative for virus, while other tissue cultured plants that had the virus are testing positive still. So it's looking really good that we most likely have, through meristem tip culture, eliminated viruses in at least a couple of the plants that we have submitted to USDA. So they're a little bit ahead of us. I'm kind of going to go back to Elisa and ask her to hurry up so we can beat them. It's not a race. It's kidding. Thanks. I don't even need the 3 minutes. I'm pretty much done. On the left-hand side are some of those plants that are virus-free currently. So we've tested them. We need to grow them out, of course, in media, let them mature, and then look at viral load or virus presence, because they could be outgrowing the virus in tissue culture currently, right? So we're almost there, I think, and fingers crossed that we will have virus-free plants for the industry soon. It's possible that we might have some only virus-free plants anywhere. So that would be very interesting. Here's our tissue culture lab at Komohana Research Extension Center. It's a little bit smaller than Lion Arboretum's. If you guys go see Lion Arboretum's lab, ours is a little bit smaller, but we're pushing for funding and for expansion of the lab because tissue culture is really important topic for Hawaii right now as a method to create cheap plant material for both the landscape nursery crop and also the floriculture industry. So Elisa, big thank you to Elisa. She's working really hard, not just on ginger, but on breadfruit, banana, taro, sweet potato, Tracy Matsumoto, John Suzuki, previous bias diagnostician, Dr. John Hu, and his grad student, Adriana Lorea Sarmiento, Xupeng Wang, Anna Luchin, did a lot of help getting us plants on Maui and touring me around, and of course Alberto Ricordi, who's working on Oahu now, helping with this project, and Hawaii's ginger growers. Thank you. Quick questions?
You know if any of these viruses are seed transmitted?
Yeah, Ken, you've asked that, and I've always said, I don't know, and then I've never. Yeah, remember, because Ken's got plants with seeds on them, so we were thinking of looking at them. We should take the seeds and give them to John, who said we can test anything, so we could try that. Yeah, but I just haven't looked at it because, right, we don't see seeds on gingers a lot.
Well, it's something compatible, but you can make the cross, and I've done that,
and that's why you have seeds on those.
No, the reason I have seeds on them is because I've got multiple varieties, that's because I've adapted, so they're self, not self, they're crossing, maybe with the help of insects. But the seeds are not part of the produce, and I used to use it like a seed fertilizer.
That would be interesting. Yeah, let's look at that. Let's look at that.
Also, I would encourage you to use, because there's a couple of really effective antiviral compounds that I use to clean up affected rodents.
In tissue culture or whole plants? Oh, yeah, okay, so yeah, I remember you did this with the orchid that we released.
The Northern Bowl on it. It's totally infected. You really make that really good success rate, like 40%.
Yeah, I'm struggling, I want them to make choices, the two labs on how they want to do virus elimination, but I can gently caress the use of some of these chemicals. I'm gonna move on then, Dwayne, or do it.
Yeah, when you say testing positive for the virus, is that by PCR or by electron microscopy?
PCR, so they're generally using PCR for these 6 viruses. Yeah.
So when you discover a new virus in this kind of work, does that slow you down while you describe the virus?
No, people cheer because then we get to write a publication on identifying a virus, but it didn't slow us down at all. Yeah.
Yeah, I heard you saying really happy that you found the virus.
Well, it's just exciting to see virus. I know it's not great, but yeah. Yes.
Plants in the habitat, are they ever infected by these viruses?
We've only really been looking at produced plants because we're focused on helping the floriculture and the nursery and landscape industries. But I would expect that Alpinia purpurata in the wild could become infected by these viruses. And there's a likelihood that it is out there in the wild. I know there's conservationists who are really hoping that it would spread to Heliconia and get rid of all the Heliconia that's invading Hawaii. So maybe for them that'll happen. But we got to move on. Sorry, folks. Okay.
Once you eradicate your field, that's your way to go to eliminate the virus?
Sure.
Does it stay in your soil, or how long do you have to?
No, the virus can't survive outside of plant tissue. So if you happen to have a rhizome that's still alive in the soil and you shove your shovel into that plant when you're replanting, and then you accidentally shove your shovel into plant tissue of the new plant, there's the potential that you could mechanically transmit it. It's not guaranteed, and we don't know the efficacy rate of the efficiency of that transmission, but it's better to ensure that you have no virus infected plant material. But it doesn't survive in the soil, and it's actually non-persistent or semi-persistent in insect vectors. So if you slow down insects from entering your ag ecosystem, they will lose the virus inside of their gut or their mouth. wherever it is, within, for most of these viruses, 48 hours. So that's what we're pushing for, is trying to reduce insect movement when you end up having virus-free plants. But we'll be doing a whole thing for you guys when we have virus-free plant material to help you keep them virus-free. And I'm looking forward to it. Yeah. All right, aloha. Oh. Give me 1 second. So sorry for going over Dr. Loges, but I would like to introduce Dr. Vivian Loges up to give her talk. Vivian is an agronomist with a PhD in botany, plant breeding, and a postdoctorate at the University of Seville. Oh, and she got her postdoctorate at the University of Seville. She is a professor of floriculture, ornamental plants, and landscapes for undergraduate and graduate students at the University Federal Rural de Pernambuco in Brazil since 1998. So welcome, Vivian.
Where is your necklace?
Oh, I don't know. Let me get you loaded up.
He's without necklace. Delay. Should have one also. Okay, hello everybody. And I have 87 slides to show. Fortunately, some of them are double because I make the translation of Portuguese to English, and some of them are double. Then I will try to be faster. And I have only 45 minutes, and later we will have the presentations of Dr. Riley. And I will try to, the name of the presentation schedule is only landscape, but I will try to demonstrate more things about the jobs, the work. Which one is?
Just left.
Mm-hmm. The work that we are doing in Brazil with mainly Heliconia, but we also work with other tropical flowers, gingers, Etlinger also. And sometimes I will make a joke to say, oh, you can look this in our bulletin that is in side. Well, when we get out here, have the bulletins, and many of our work is in this bulletin, and can want to take that we take all of them away. Then I will present Zingiberales in Brazil, from landscape and cut flowers to use as a bioremediation plant. It's a work that I do with my students and many colleagues that are very important to me. And as you know, Roberto Burle Marx was the first one to introduce tropical plants in Brazil, to use our plants in Brazil, our Heliconias and other species, and bring a lot of species from outside of Brazil, changing materials with other farmers. And he's a famous landscape designer. And he said, use and transform the exuberance of the jungle to use in landscape. And this is a few Heliconias that we have in this place. In this place here, near Amazonia, and this is in the middle of the jungle. Then we have, I'm upset that we are not the hotspot of Heliconias. It's Colombia and Costa Rica, but we have some of them. And when we start to study Heliconias to use as cut flower, many of our farmers like to make a joke. Oh, I have 122. Oh, I have 123 different Heliconias. And they bring very different types from the jungle to produce as cut flowers. And we know a man that comes from Amazonia and bring this Heliconia and said, I have a white Heliconia. And everybody that have always red Heliconias, yellow Heliconias, want to have a white Heliconia. But it's forbidden to bring material from north part of Brazil to northeast part of Brazil, because they have disease, virus, and many diseases that are, because of that, I always frightened to take something to my country, to bring some disease that is worse for our bananas. Bananas, unfortunately, are most important, very important, most important than our Heliconias or other gingivias. Then I don't never bring materials from my country because I'm afraid to take virus from Alpinias or something like this. And then we started to make those studies with Heliconias, and only to localize you. I live in this part here called Pernambuco, the richest part of Brazil, the southeast part, where is São Paulo, Rio de Janeiro, and where they produce with high technology. We have a veiling that is similar to Aalsmeer in Netherlands. We have a lot of different types of productions, mainly for temperate flowers. And in northeast part of Brazil, where I came from, this is here, Pernambuco, almost the nearest part of Brazil from Africa, is where we produce tropical flowers. And many years ago, we started to produce tropical flowers, imagine to export them, but it's difficult. They are too heavy. We see this later, and then when we started our research, now we see that we need to improve a lot those tropical flowers to make exportation. And this group, my state have different climate conditions. We are a little bit poor, then we have low power consumptions, then we send flowers to the south part of Brazil. We started many years ago. Our productions are around the main city, that's called Recife, and now we produce to local and national consumption. We have been the first national producers of tropical flower, but now we are few of our farmers producing Heliconia. And we try to export, we export Heliconias, but now anymore. We see that it's easier to export inside Brazil because when we take a flower from this part of Brazil to the southeast, it's like it goes by airplane, and we take care of the flower as we are making, exporting flowers. You are so silent. I don't understand it. Wow. Thank you. You understand? Yes. And then we start this research about the Heliconias. Okay, 2 minutes. And we start talking about this is a good business for small farmers who the heights at the end of the products. And we have a few small farmers, but most of them are high-level farmers producer. And currently we send our leaves and flowers to south and southeast regions of Brazil, where we can they consume and pay high values of our flowers. But it's 10% of the price that I see here in Hawaii. It's very expensive, the tropical flowers. And here is our local market with tropical flowers. And we make a group in 2005, with our group of 4 that came from different institutions, 3 different institutions of different parts of Brazil, and we start to study mainly Heliconias. This is Carlos, who everybody remembers Carlos, that organized our meeting in Brazil, and Cecilia that is from Embrapa. Unfortunately, Embrapa is our research center of plants. They are not working anymore with flowers, only with things that we can eat. No more flowers, it's not important, and we are not studying anymore. And this is another colleague of Carlos that is works in this institution of São Paulo that is now there. They are cutting the money to work with flowers also. It's a pity for us. Then we start a pre-breeding program. We don't make process because we have already so many different types of Heliconia that we decided to characterize them before we imagine to breed them. And those characterizations, I now is wrong, it's not 200 species, it's 187 species. The mistake. We start to look from this diversity that we have with the farmers. As I said, they have 100 and 122, 123 different species. And we started to make research with those species from the farmers. And looking from the position of the inflorescence, type of inflorescence, color, hairiness, waxiness. When we started this research, the producers are not producers, they have a collection. Nowadays, they throw away all the species that they don't want anymore to use as cut flowers. It is a pity for us because we lose our materials. And many materials that come from other countries. Então, which species to grow as cut flowers? And this is one that in this Portuguese, sorry. Sorry. Don't forget this one. If you want to learn Portuguese. Okay. Okay. Then we make a morphological and agronomic characterization of 52 genotypes to select cut flowers during this period, almost 10 years. I had a lot of students, slaves, which and one of my inspirations was Dr. Crowley, that I read the works from Dr. Crowley and I follow the same methodology that Dr. Crowley used to characterize plants, to count shoots and everything. Then in this material, we saw the size of the clump. And one student of mine, we saw this part above, but we saw no, the plant grows up also. Then we need to see both the projections of the clump and also the base to decide which space to use from these plants. And as Dr. Crowley wrote from Heliconias, you have open types and grouped types of Heliconias, depends on the type of the rhizome. Our conditions was open and without protections, only from trees. And we have two systems of irrigations. And we look to see if it's annual seasonal productions of flowers. And this is what we measure: height, plant habits, plant base, and projections area, shoots in the internal part, inflorescence number, and bending down tendency. We measure that it's difficult when the shoots fall, and you need to maintain them to pull them up. Another one in Portuguese. Sorry. We start with one rhizome per plant. And count each shoots that each rhizome developed during 5-6 years. My students, students, yes. And we developed this methodology based on Dr. Crowley works. And then as we measure, since the shoots come out, we have a tag that we put when the inflorescence get out, and then we have the cycle of the plant, how long it takes from the shoots to the end of inflorescence, and when we cut the inflorescence. And then we measure all the inflorescence, the length, the diameter, the fresh stem. Some of them are very heavy, and the durability in the vase, because we need to know that we have more than 7 days, 10 days. It's too short, the durability, the post-harvest durability is not a good one. And we have these two types of development and grouped or open. And this is a photo that I take in Australia from Marianne and my farm, the producer of Australia. I forget. Maureen, and I think it's very interesting that to use a manage the clumps in a different way. She cuts all this side and lets this side come here. And she cuts the other side of the road and lets the productions walk from the bed to the, I said this in English, from the the production space to the space between the clumps. And in Brazil, we remove everything from the soil and start it again. Then you stay in a very, very low number of inflorescence. You get zero and to start again to produce. In this way, you maintain the productions when you maintain the shoots coming to here and cut these shoots. Because it's necessary to remove and renew the production. 13 years. I think I'm going. If you have any questions, I can answer one question. And then we take almost 10 years to select those criteria to cut flowers, to species of Heliconia as cut flowers. And the amount of shoots for plants, the amount of flowers for plants when they start to produce. If we see that we have shoots that die, we observe that many shoots only stay still green, never produce flowers. 10% of 100 shoots only produce flowers. Of one species like Heliconia nappy yellow, this one here, the production is very low in our conditions of Pernambuco. Inflorescence cycle, inflorescence aspects. This was a criteria that we adapt from Alpinias to Heliconia. Type of inflorescence, size of inflorescence, presence of hair or wax, presence of flowers inside the bracts, because we associate the presence of flowers inside the bracts, and if there was upright species of Heliconia, that the water became inside and the flowers smelled bad, bad smell. And I had a student that found the insects inside the bracts of the she worked collecting insects of 20 types, different types. Oh, this is written in our bulletin. You can find out the bulletin, and this is a work that we are in the bulletin. And then we saw these insects to control, to start to find out methods to control those plants, those insects in the plants. And when we are happy, we have 26 different characteristics to select. This is suggested to follow. Those ones we suggest to follow. Oh, sorry. And those ones we should not recommend it. I take those characteristics to a farmer that I work with that always help me and always co-author from the works that I present. And she said, no, why so many different characteristics? I only follow those characteristics, selected 3 or 4 that we work with. This farmer is called Maria do Carmo, is one of our famous producers of tropical flowers. And she used to pay more $200 for rhizome. Imagine, they fight each other to have rhizomes. It's true when you know where the rhizomes, the special plants are, someone could go there and stole your rhizomes. And then because of that, you must hide the special types of rhizomes. And now she selected few. She removed all the plants species that should don't are not enough good to produce flowers, and they have not more a gene bank, but a commercial producer. They remove all of those plants. And then she explained to me one day that I arrived there. Oh, I select Heliconia like this. 3 starts to productions along the year and market accepted. 3 starts to long-lasting for harvest durability flowers species. 3 starts to productions during May to July, when is our scarce period of inflorescence in Pernambuco. Then she gives 2 starts to Heliconias that require low maintenance, 2 starts to better marketplace. And I imagine that if this is important, how is easy or not to package the inflorescence in box, but she gives only 1 start to this characteristic, and novelty aspect of inflorescence on one side. Then all the things that I imagined that is important, she said, no, this is important to me. Only this. Then in my table, those are important to the farmers, and those other one is not, sorry, those other one is not so important. Then it's important when we are researchers to stay together with farmers to see what is really necessary. And we stay from 10 years study each shoots that come from the floor, counting and looking which one became a flower. And she have this table. Oh, in January I sold all my sassy inflorescence. And I with this table that she put here, I have flowers to sold, I have here less flowers to sold. She knows now the seasonality of these species. When peach pink here, she don't have how to produce during those months. It is a farm that sold all the flowers, and then she counts how many flowers she could sold per month. Then she had this table that she can follow the seasonality of those plants in Pernambuco. Nowadays, she's retired, yes, and his son are working, our son are working now in the farm. They are always giving up of Heliconias because when compared to alpinias, have less flowers per square meter than alpinias have. And they are now working with leaves, Cordyline leaves, Lupi bamboo leaves, plants, more than those orchids of Heliconias because they are not so producer than other species. So just to show all the works that we done in our lab, we characterize chilling injury because we saw that sometimes the person confuse the symptoms of senescence with chilling injury. And then we have a work that we put the flowers in the refrigerator and take them there, frozen, to see the difference of the chilling injury that get dark marks like this, or the senescence that when the drugs get dried. Because sometimes we have problems when we send flowers by flight, and they don't put in the right place in the airplane, and then we get low temperature during the flight. This is the Heliconia lobster claw, and those Heliconia cultivar Halloween. They present different symptoms of chilling injury, but it is diverse from senescence. There was another article that we published in our collection. You can look at this because I have the photos from all many different species that we demonstrate the difference between chilling and senescence injury. Then, the subject of the presentation, when we look at the plants as cut flower, we start to say, why so cut flowers? Why not ornamental plants? Because we start to use Heliconia as cut flower, not as ornamental plants. And then we saw that most of the information that we have looking from them as cut flowers, we can use as ornamental plants. And many of the species that is not good enough as cut flower are wonderful from ornamental plants. And then we saw the color, the shade, the arrangement of the bracts, the length, the number of the bracts, if it has bad smell, and the visibility of the inflorescence. We have some Heliconias that we need to move all the jungle in front of all the leaves to see the inflorescence. And we're very concerned about the occurrence of insects because we have diseases that are transmitted by insects, and the durability of the plant in the clump. Okay, and as cut flower, when we use the plant as cut flower, you cut in this point, but in ornamental plant, you can leave the plant for many more days and let the bracts open. And this was what we call cycle of inflorescence in the plant. For example, Then we use the same table to select the criteria as ornamental plants, adapting from the criteria that we use from cut flowers. And for us, well, as I explained before, the visibility of inflorescence, the ornament aspect of inflorescence, the requirement of maintenance. We thought that hair on the leaves or rocks will be a problem, bright colors that will be usual or vibrant. And this is for us important, the black dips, and the possibility to have an aquatic habitat and have insects inside. And we are afraid of mosquitoes inside the blacks. And we hope we don't find mosquitoes because we forbidden us to export flowers from anywhere. And then we do for example, Heliconia niqueriense, we can use like this as massive plant. It lasts 27 days in fluorescence in the clump, covered large spaces, and you can use along the walls or the fences in public area and have a rustic and low maintenance requirement. And with them, they just work from other inflorescence in a place that we work in São Paulo. All the explanation that I've done is northeast part of Brazil, where the climate is almost like here, 30 degrees, 38 degrees. But in this part of Brazil, that is in São Paulo, it's a little bit much cooler. And I think it's the place with the highest amount of rain that I have ever seen. Rain very much in this place, very near the sea, and we have been there when the Heliconia meeting was in Brazil. And then we do the same. We follow clumps and inflorescence of many 10 different Heliconia to say how the height of the plant, leaf to the base, the height of the inflorescence. I remember that I have been in the Carla's house, and I saw the highest Heliconia that I never imagined could exist, 10 meters. Take a picture of mine, and we even can see the inflorescence because it's very big. I think it's not good to landscape because we need to stay like that to the inflorescence. Even I need to see the inflorescence. And when it's in our view, 2 meter high, 1.5 meters, 2 meters, 1.5 meters is okay. And keep flowering from 3 months. The inflorescence is very interesting. It walks 60 centimeters of length and lasts in the clump 100 to 150 days. We will see many inflorescences where have always those information. I'm not sure if this work is in the bulletin, but if it's not in the bulletin, we'll try to do. We are trying to convince you to take all the bulletins that we have here and become partner of our society, looking for our articles. Another one, Policiana, I go back. Okay. Pauliniana, it's a crazy one, the size of the plant, the way that the bracts open, and the size of the plants, the period, it have a lot of seasonality for us. Again, details of the inflorescence, all these information are in the articles. And the interesting thing that we saw is that we have Heliconias that require more maintenance to remove old leaves and to keep the appearance better. And one thing that I most explain that we are discussing about is about Heliconia cartacea. This place here is in São Paulo, and this clump was forbidden. No one take care from this clump. And it's full of flowers. No one could fertilize nothing. And I started to do a work with a colleague, with a student that have a plantation of Heliconia cartacea that was also abandoned. Yes. And we go there each 15 days to remove inflorescence. And after 6 months, all the plants disappear. It's like they always die. It doesn't happen with Heliconia bi. We imagine that's because within Heliconia bi, we have, in our place, we have many shoots that only stay green, don't have flowers, vegetative shoots, don't produce inflorescence. In this place that we have this Heliconia, we imagine, we are not sure, we suppose, each shoot became a plant with inflorescence. And we remove this from the soil to soil. And then all the reserve of this plant don't go back to the rhizome to produce another shoot. And then they don't have how to maintain the plant. And this is an interesting study to do who wants to work with cut flowers. If have plants like this that we have, we heard only about the Heliconia cartacea. No other species we heard that we have this problem in our country to die because we remove all the shoots. Because all the shoots became inflorescence. If not all the shoots became inflorescence, I leave them there. I will not remove and they maintain making the photosynthesis and produce a new shoot. So because all is connected by the rhizomes. This is very interesting plant, one of our most interesting species from Amazonia, and we are not using them as cut flower. Okay, I will give more info. Photos. All the platystaches I think we saw here is very interesting. This is very rich for me. My size of inflorescence, 7-meter plant with those pogonantha with this very big inflorescence, only as ornamental plant. To use this cut flower, I think it's too heavy, the steam, and we're forbidden to transport them. And as it have a lot of, it's not us also as good from cut flowers because this dark part in the bracts began very early in inflorescence and looks like senescence, and the person will not like. But it lasts many months in the plant. Velerigera is interesting also with these hairs. 5-meter plants and 4 months of inflorescence for blooming. Inflorescence with almost 1 meter and 30 centimeters is very beautiful one, and the flower lasts more than 150 days. Another detail that we saw here. Oh. Spoiler. I need to go faster. And I'm not so fast. I can come here. Sorry. I think it's my hand. Okay. No, I'm looking here. Okay.
E agora, onde é que
Please speak slow. I have plenty of time. Okay. No, I will keep this.
No, it's only, she has it for
Oh,
PDF, so it's
okay.
only up there. Yeah.
It's mine now.
I can see that. Okay, and then this is pendula is not so beautiful plant. We have a lot. Oh, sorry. Oh, Rostrata. I remember when I read the first article from Dr. Friday, let's say Rostrata 3 days and Rostrata 10 days. And what is that? 3 days and 10 days. Oh, then when we harvest those Rostrata, one lasts 3 days post harvest. We have only 2 days. The other one 10 days. And I always teach to my students, if you grow Heliconia rostrata, which one you grow, the 10 days or the 3 days Heliconia rostrata? I imagine that we must produce the 10 days Heliconia rostata that you can sow far away or from a short event near your place. And one day when I visited the farmer and I asked, oh, of course you only have 10 days Heliconia rostata. No, I have also the 3 days Heliconia rostata. When I know that is from a short meeting, a marriage, something like this, I sold the 3 days rostata. When it's a big meeting, something that lasts more than 3 days, I sold the other one, because for us, the 3 days Heliconia rostata produce more inflorescence than the 10 days rostata. And then we have both of them. Another problem was that when they sold to the farmers, no one know that no one read your article, Dr. Private. No one know that we have 3 days less and 10 days less Heliconia rostata. It's one of the most wonderful from landscape. I remember when we visit Indonesia, we walk in the House of Pan, a place that have Heliconia rostata that is very interesting. Pendula I don't like so much. It looks like ears of animal, but it's a difficult plant to maintain. Yes, but it's interesting. It looks like, yes, but it's interesting also. And it depends on the taste of each one. Then we can love all Heliconias. It depends. Even the ugliest Heliconia that Carla make a presentation, that should have done a presentation here. There is a plant that's full of sand. Yes. I have the presentation in your photo, but I imagine that we will show them. And these episcopales, I'm very surprised the connections of episcopales with other Heliconias, and when they are from so different parts of the world, and it's so different Heliconia. If I make a breeding, I think is interesting. Make a breeding of Heliconia episcopalis and shy like this, and Heliconia laxpata or other Heliconia that is like this that needs many space to put in a box. Pauliniana, we call crazy one, lasts many days in the clump, and despite the seasonality, she can start to flower in a month and produce many flowers that you almost have flowers all the year. It's a very interesting ornamental plant. Then we have many Heliconias that we can use as landscape plants, and we need to look for plants, spot plants. And then I move my research to other type of subject, and I start to work with nature-based solution and start to work with green walls. And I joke that everything that passed in front of me, I taste in a green wall. And then we have this plant here that we call ear of elephant. That is very big, it's one aracea, and we put in a green wall, and it is very huge. And I said, I can look when we walk around, and why not Dove Jamaica as a plant to a green wall also, because it's very small, the smallest, I think, so Heliconia that we have, that we can also use in green walls. And I stopped to study Heliconias for almost 5 years. And then we start again to study Heliconias to use as green covers on nature-based solutions for urban planning. And I work with green walls, I work with green roofs, and Passiflora also was green cover. And during the pandemic, someone invited me to help to choose the plants to use in a rain garden, the drainage garden or rain garden. And what I do, I look around the place where I work, and I saw places there that when rain are full of water, and when we have periods of dry, there is no water. So this plant this land can be used in these conditions of rain garden. I'm sorry, I don't remember if we don't use costus, but costus will be very interesting to use in this situation. And also, we start to look for filter gardens too. Then I began to study again heliconias and tropical flowers, because I'm studying grasses, other kind of plants, and we develop now studies with those type of gardens that I translate as rain gardens. It must be a bipolar plant. Imagine, I can be without water and I can be full of water. I can be without water and with a very amount of water, because when rain, all the water are conducted to those rain gardens. And then we start to select, this is the garden, that the first one that we have done, we use Heliconia VI and also Cana indica, and Alpinia. I'm also without views, and Alpinias, and also Lamanda. They are now developed alone, each one, some of them disappeared. But Heliconia VI for us was not a good choice, because it's very invasive and the rhizome is very big. And the idea is to use those systems in streets, calçada, where the person walks inside, yes. And then we cannot have a huge plant, because we will not see this. But to be a nature-based solution must be native plants. Then it's difficult to find all native plants to do those base solutions. Then explaining a little bit about rain gardens is to manage the surface runoff, cleaning the pollution that runs from the surface, protect the water resources, since you filter those water, restore the vegetation and biodiversity. As you have a garden, you have a lot of this in the United States. Habitat for wildlife, and increase the moisture of adjacent soil. And the most important criteria, okay. The most main selection criteria that we used was resistant to those big model of water, without water and true. And cannot be so large species. Though I guess okay. Then my students developed this type of base to have the plants that have these soil types, and when the water get in, stay in the soil, but most of the water go to where we have the gravel. And topsoil, washed sand, and coconut powder we use as material to plant the plants. We work with those species, Stricta, Episcopalis, Laxata, Rostrata, and we evaluate many small ones, Citacorum, Richardiana, in our climate, it's not a good plant, doesn't accept it so hot weather that we have in our states. And we have donations of the rhizomes from the farmer that we work with, because we saw that it could be a potential way to receive money selling those rhizomes to plant in those type of garden, and then they give us and give us to the rhizomes to make those research. And the experiment was in shade from 90 days until the plants start to grow. We daily, in the beginning of the experiment, water the plants, but after we maintain the plants in dry situation and compared to other part of the experiment that we put water all the days. Then when we make this 15 days without water and 1 day full of water, we imagine that this is what happened with us when we had rain And all the water goes to the rain garden. Many days without water and a few days with a lot of water to see if the plants support those type of management. The best genotype for us was Heliconia niteriense, Piscicola and Sassi. They establish very fast, have a vigorous growth, bloom very early, have a good durability of inflorescence because it will be interesting for landscape, the inflorescence maintained in the plant. And we observed that many plants have leaves that get dry because of this remodel type of maintenance without water and water. This is another experiment of other students that are using rainwater to grow heliconias and to sow the inflorescence. They like very much the rainwater and that produces very well. And my city approved financial support to construct and pass to construct a system of a filter garden. We have a problem of our house don't have, maybe a poor person in Brazil don't have water where to go. Sanitary system, we don't have many houses. We don't have sanitary system. Even buildings. Yes, and then the dark water go to channels and the channels go to the rivers. And then we have money, financial support to build these filtering garden. And this filtering garden to be a nature-based solutions needs to be with native plants, and they use heliconias in this native garden. This is the place and where it was built. You can protest.
No, passou.
Okay. And it's like this that they build this garden. Sand and gravel. And they plant the heliconias and the plants in gravel, right? Gravel? Rocks. How can support? But it's almost a hydroponic system because they maintain the water with a lot of nutrients because it's organic water, not clean, organic. And the plants are perfect there. And they have two systems, two gardens, one that the water come like this, horizontally, no, vertically. And after pass, vertically go to another garden that the water pass horizontally. And then it's too expensive to see the pollution in the end of the system, but the water arrive dark and get out very clean. And this is when they planted those plants that they use from the from the Zingiberá family, Canna generalis and Heliconia psittacorum was the one that we use in both gardens. This is the vertical filtering, then the water falls in the gravel and after goes to the another and milk. The plants are perfect. This is the horizontal filter. It's the same system, but the way the water passed during in this garden. And the most interesting thing that I observed is the water is here in the plant, not here in the shoots. And the boy that take care from this place, so he said, one day I asked, how long that plant become full of water? 10 years. I said, no, how many days? 10 years. Can became in this kind of garden with full of water. But what they do, they have two systems and they change. Because if I maintain water, produce musgo, produce this yes, and then they dry it to don't have soil. This ring that cover when we have water in the sand, and work with this system and change to another system and work like this. Then each week work with one of the two systems. And no, and the most interesting thing that I observed is that they each month, each period of 6 months, 9 months, they cut all the plants on the floor. If you are looking, this man is cutting all the plants. And for my surprise, they bloom, they grow again and bloom. And of course, they bloom at the same time. Because when you cut everyone, and each shoot will produce almost in the same time, you will have flowers in the same period. This is a problem that I talk with my students, how we can solve this problem to have flowers in the Mother's Day or in Christmas. We cannot do this with Heliconias, but looking the system, they they are not looking for this solution, but they show us the solution as person that cut flower, the farmers cut flower. But it's different system. In soil, I don't think that's the same. Because imagine all that the roots are taken from the dark water that they receive that are full of and this is interesting. It's already used in Mexico and another country from South America, from Central America, to clean water from pig creations, from person that have pigs and clean the water. Pigs. Pig farms, pig farms. Then, as you can see, this should stay like that. This is the last part of this pools where the water became oxygenation for the oxygenation of the water. I'm finishing. This is the view of the top of the place. For us, it was a gift to have now this place to visit, and an inspiration to do that in our house or in each space, because you can do this anywhere. Oh. Over there is the water, how it arrived and how it goes, yes. And the river is very beautiful. We call our city Veneza Brasileira, meaning Brazilian Venice. This now is out. And our commercial time, please wrote to our Heliconia Society, anything interesting, don't be shy. We have very, very interesting articles, like my friend sometimes sends something funny, Dr. Press, when they the travels around the world, Dr. Friley sent many articles to us. Now I'm responsible from the bulletin together with Ken. And please go to the website or take bulletins. Don't go to the website. Take bulletins. And we can make our society. Each one is not that. But don't go to the website. Sorry, I don't know. The bulletin will be there. Don't go to the website. Take that. Yeah, we have, I need to do the same you have done, an index. I saw that the last index was done in 2015, and I'm now in 2025, I will need to do an index. But we have an index from 2020 to 15, where most of those works that are present are there. Then you can find that. And thanks to my colleagues and my student slaves, this is great. This is the photo where our meeting in Brazil, many of you are there. This is Carlos that organized, my friends, and my students. And this is only possible because of the financial support of the societies that are here. Thanks from the Heliconia Society, and thanks, Russell, from all of your work to make us be here. And let me speak 52 minutes. Okay?
Question?
I can make a question to myself.
I just tell Vivian that I have a costas for her water garden. Oh, Costas Vicearum, because it grows in Vicia flooded forest. You can go in knee deep water, the flowers just sitting above the water.
Vasia is the name that we use to a place that is full of water, wetlands.
Seasonally.
Seasonally, yes. Especially by the sea or the Vasia. I think I explained everything, no questions?
Cost us for every occasion. Thank you very much, Vivian.